This is the total flowchart on insulin action. Action of insulin. আমাদের এখন কি চলছে? Endocrine pancreas. Last week we have started endocrine pancreas. Simultaneously, as you are writing it down, we'll be recapitulating a little bit of endocrine pancreas. Pancreas, as you know, it's a mixed gland. And since it's a mixed gland, it has maximal majority part of the pancreas is the exocrine part. Exocrine deals with digestion. Digestive enzymes comes out of exocrine part. Endocrine deals with hormones. Hormones er moddhe there are multiple hormones like insulin, glucagon, somatostatin. Individually, these hormones are secreted from specific cells. Like insulin is secreted from beta cell, glucagon secreted from alpha cell, somatostatin secreted from delta cell, and there is also a very interacting relationship between these two because all of these cells do have paracrine effect on each other. So alpha has paracrine effect on beta, beta has paracrine effect on delta, delta has paracrine effect on alpha, like that. This happens. Clear? So on the picture, on board what you are seeing, this is insulin. The majority of insulin action is on carbohydrate metabolism. The major part of insulin action is on carbohydrate metabolism. But it has significant effect on protein metabolism and significant effect on lipid metabolism too. Whichever is in the blue box, that is lipid metabolism. Whichever is in the yellow box, that is protein metabolism. I'll explain. And whichever is, without any box, out of the box, that is carbohydrate metabolism. Okay. So I'm giving you another 1 to 2 minutes for quite a few of you are writing, and then we will be moving into one after another effects, basically. If we look at the gross effect of insulin, its major effect will be main target. Insulin main target is to reduce glucose in blood. So overall reduction of glucose in blood. So reduction of glucose in blood can be achieved by a number of factors. It can be like that insulin may stop glucose exit into blood. That might be one thing. Insulin may convert glucose into something else and store it somewhere else. That can be another option. So এই পুরো জিনিসটা সেইভাবে আমরা করেছি। If we look at, এখান থেকে আমি এবারে এক্সপ্লেনেশনে যাচ্ছি। একটু টাইম নিয়ে যাদের ছবি এখনো তোলা বাকি আছে নিয়ে নিতে পারো। Then we will move into the explanation. আরেকটা অপশন হচ্ছে you can take a picture, later you can write, that will also help in practice. Now we are moving into explanation. Insulin here, what is this? GLUT4 dependent entry. I have written GLUT4 dependent entry. So insulin acts in such a way that whenever insulin level is high, glucose transporter GLUT4, there are multiple types of glucose transporters. Last week we have discussed, of which GLUT4 is insulin dependent. So what happens under such condition, GLUT4 is expressed on the cell membrane. So whenever GLUT4 is present, দরজাটা যেহেতু ওইখানে আছে ওটা খুললে লোকে ঢুকতে পারবে। GLUT4 section is just like that. GLUT4, this is a protein and this is present in the cytosol. Whenever favorable environment is coming, there is a series of phosphorylation and activation of these protein molecules. These protein molecules then they will be transported to the border of the cell and they will open the gate depending on whether insulin signal is coming and whether glucose is adequately available. So whenever glucose is adequately or highly available in blood sugar, in blood, in that case this GLUT4 will open up, glucose will enter into blood, enter into cell. Glucose যদি সেলে ঢোকে, ব্লাডে গ্লুকোজটা কি হচ্ছে? কমে যাচ্ছে। So that's the function of insulin. It will reduce the blood glucose. That's the function. One point is over. Increase uptake of glucose into hepatocyte. So hepatocyte কি হচ্ছে? Hepatocyte glucose entry is to some extent insulin independent. আগের দিন বলেছি, because GLUT2 is acting over there. So যখনই গ্লুকোজ বাড়ছে, through GLUT2 inside, so blood গ্লুকোজ বাড়ছে, glucose increased. Hyperglycemia is happening. Hyperglycemia means glucose high in blood. So whenever that is happening, glucose is moving into hepatocyte through insulin independent transporter. তাহলে আমি ইনসুলিনের ফাংশনে কেন এটা রেখেছি? The reason behind this glucose transport into hepatocyte happens due to gradient of glucose. তাহলে হেপাটোসাইটের মধ্যে কোন জায়গাটায় লিখবো, হেপাটোসাইটের মধ্যে ট্রান্সসেলুলার যে গ্রেডিয়েন্ট হচ্ছে গ্লুকোজের, ডিপেন্ডিং অন গ্লুকোজ এন্ট্রি উইল হ্যাপেন। সো ট্রাই টু আন্ডারস্ট্যান্ড, সাপোজ দিস ইজ হেপাটোসাইট, inside hepatocyte glucose is present, outside in blood glucose is also present. Here which do not require insulin for the entry of glucose. Now when will glucose enter hepatocyte? When outside glucose is high and inside glucose is low, depending on the gradient. Insulin's action is glucokinase, and glucokinase ta ki kore? It increases the glycolysis. Glucokinase will break down glucose. So the more glucokinase will work inside hepatocyte, glucose level will go down. And as that is happening, cellular glucose gradient is high, glucose from blood will get into hepatocyte. Again we got that glucose level in blood is low. That's the target. Clear everybody? So এইটা হচ্ছে আপডেটটা হচ্ছে that way it is working. Third point here, this one. What is this? Glycogen synthase. So কি হচ্ছে? Insulin stimulates glycogen synthesis. So what will happen? Glycogen synthesis will go up. So what will happen? Muscle glycogen and liver glycogen both will go up. So the storage form of glucose is glycogen. So if glucose is stored more, তোমার মানি ব্যাগে টাকা আছে, আলমারিতে টাকা আছে, তুমি যদি বেশি টাকা আলমারিতে দিয়ে দাও, মানি ব্যাগে টাকা কমে যাবে। That's it. That's the storage. So storage form of glucose will be up, the free form of glucose will be down. ওটাই ইনসুলিনের কাজ। So blood glucose will go up if glycogen is high. So that way it is happening. So next, তার উপর একটা insulin inhibits glycogen phosphorylase. Glycogen phosphorylase is the very important rate limiting pathway in the glycogenolysis. তাহলে যদি glycogenolysis means what? Glycogen breakdown is happening. ব্যাংক থেকে টাকা আবার এফডি গুলো ভেঙে নিতে হচ্ছে, ব্যাংক থেকে আবার টাকা তোলা হচ্ছে, সেটাকে if we can block, what will happen? Free money will be down. So glycogen breakdown is inhibited by inhibition of glycogen phosphorylase. ফলে কি হচ্ছে? Blood glucose will be low because storage form is maintained. Clear everybody? হয়ে গেল? Phosphofructokinase and pyruvate kinase. Insulin stimulates or excites or stimulates or switches on phosphofructokinase and pyruvate kinase. What does এরা কি করে? Glycolysis. So what will happen? Like glucose is there in blood, and since glycolysis is happening, glucose is broken down. Carbon dioxide হয়ে গেল, lactic acid হয়ে গেল, pyruvate চলে গেল, whatever, but glucose is not there in blood now. So overall glucose level in blood will be low. Clear? Glycolysis হওয়ার পরে এই জিনিসটা important. Insulin enzymes I have written. More in biochemistry you have to remember this kind of things, but still here one is pyruvate carboxylase. Insulin inhibits pyruvate carboxylase, glucose 6 phosphatase, fructose 1,6 diphosphatase. Our অসংখ্য enzyme আছে, just the important one I have written, and these are the rate limiters in, and these are the important enzymes in neoglucogenesis. What is neoglucogenesis? Something that happens in liver. Neoglucogenesis is important for hepatic glucose output. Liver ta bhishon important. Only pancreas diye sugar control hobe na. Liver is very very important. Hepatic glucose output. Neoglucogenesis. Neo means new. New glucose, that means non-carb source থেকে carb source, carb result, that is neoglucogenesis. So non-carbohydrate substances, protein থেকে হয়ে যাচ্ছে glucose, fat থেকে হয়ে যাচ্ছে glucose. So non-carbohydrate substance থেকে glucose conversion, that is neoglucogenesis happening in liver. If that keeps on happening, blood glucose will go up because liver ultimately a time will come, liver will continue the output of glucose. That means that glucose from liver will go into the circulation, blood glucose will go up. So if we can block that process of neoglucogenesis, capillary or level will go down. If we can block down the neoglucogenesis, blood sugar level will go down. There is something called Randall effect, হচ্ছে competitive inhibition or competition for substrate between free fatty acid and glucose. Muscle eta hoy Randall effect. 1960s people used to think that that is the only reason why diabetes happens if Randall effect is not proper. Philip Randall was the scientist who also got Nobel Prize for this discovery. So jeta hoy je there's an idea that free fatty acid and glucose in muscle both target the same substrate. Okay, so if we can increase the free fatty acid utilization, so glucose will not get the substrate. Kind of that's the idea basically. So Randall effect, just ekhane eta mathay rakhbe, this is so basically it's a reciprocal relationship between FFA and glucose, and insulin behaves in such a way that ultimately the glucose utilization increases and blood glucose reduces. That's Randall effect. এর থেকে বেশি এখন তোমাদের খুব বেশি লাগবে না এর বেশি. So this is more or less all about carbohydrate metabolism which is dependent on glucose. Once again, let us recapitulate. There are two very important transporters. In last week I also mentioned, one is GLUT variety of transporter, the other is sodium glucose linked transporter. These will keep on coming multiple times, so that's why we are repeating that part. GLUT transporters, these are facilitated diffusion, they don't require any ATP, whereas this one, this SGLT2, they are secondary active transport, so they require ATP. But their ATP is usually comes from the primary active transport on the basolateral membrane of the cell. It may be intestinal epithelial cell, or it may be renal cell in the convoluted tubules, and that is sodium potassium ATPase from where the ATP supply is happening. That's why glucose comes in, sodium comes in, sodium goes down on the other side. So there is always a transmembrane gradient of sodium present, which keeps on inviting glucose into the cell, and that way the glucose reabsorption happens. That's the SGLT. There are two variants, many variants are there, but for your understanding, SGLT1 and SGLT2, these are most important. GLUT also have a multiple variant, of which GLUT4 is insulin dependent, present in skeletal muscle, cardiac muscle and adipose tissue. GLUT2 is present many places, we will come to that basically. It is also present in pancreas, in liver cell, multiple areas GLUT2 are usually present, and even in kidney it is there. GLUT5, that is something which is present in testicular area, it is present in intestine, it is for fructose transport, GLUT5. GLUT1 is there, GLUT3 is there. We have found various forms of GLUT in placenta, we have found various forms of GLUT in RBC, in brain, because glucose is the energy source. Another energy source is ketone, but glucose is the energy source. So all types of various channels must be present in various areas. Okay, clear? Now coming to other parts, protein metabolism. all, insulin increases amino acid entry into the cells. So amino acids, these are the building block of cells. So if we can make our cells more enriched with amino acids, cells will grow, multiple functions in cell that will happen. যে কোনো কাজের জন্য যে phosphorylation হচ্ছে, protein kinase কাজ করছে, we need amino acid for those things. So cellular development, cellular growth, cellular signaling, cellular transport, everywhere we need amino acids, and insulin helps to transport amino acid into the cells. That is one point. Second, protein synthesis, because amino acid is getting into that cell, and further, post translational modification and everything, everywhere the action, indirect or direct action of insulin is present. And the last one is the insulin reduces the protein. Effect of insulin is anabolic. Net effect of insulin is anabolic. That's why the people who are on insulin, they occasionally, insulin for diabetes, they occasionally complain of weight gain. Explain why question. People who are taking insulin occasionally complains of, or occasionally means most of the time, or they will complain. They have to complain about weight gain. Okay, so people on insulin, they may face weight gain. The reason is this. Why may? Because this is not the only hormone. There are multiple other factors are working, that's why. But if insulin is there, and if no other counter regulatory things are happening, definitely there will be weight gain, because insulin is anabolic. One reason of anabolism is this. The other reason of anabolism is glycogen synthesis. Glycogen যদি বেড়ে যায় muscle, ওজন বাড়বে তো। Liver has more glycogen, muscle has more glycogen, your body weight will go up. Muscle will be bigger if you have more glycogen stored. Muscle store is getting replenished. So amino acid ঢুকছে, glycogen ঢুকছে, glucose ঢুকে glycogen হয়ে যাচ্ছে. But definitely there will be weight gain. Insulin is anabolic. Third one, this is very important. এই জায়গাটা ভীষণ important. Lipid metabolism, this blue box. Lipid metabolism, তার উপরে কি করে ইনসুলিনের কাজ হচ্ছে? The first is insulin reduces, or I'm so sorry, insulin inhibits hormone sensitive lipase. Hormone sensitive মানে কোন hormone? This lipase as you know, lipase is a hormone for breaking down fat. Now certain hormone acting on this kind of lipase and activating them. So which hormone? There are basically two hormones, steroid hormone and glucagon. Steroid and glucagon, they usually, এটা echo না আঁকলে it will make it very complex, তোমাদের যে ছবিটা. So basically this hormone মানে steroid and glucagon, they stimulates this kind of lipase. So lipase বাড়লে কি হবে? Fat breakdown. There will be fat breakdown. What insulin does? Insulin inhibits. There will be no fat breakdown. Again, this will help in anabolism. Fatty tissue, that's a store of things. If fatty tissue broken down into free fatty acid means কি? ওটা ব্লাডে চলে গেল, and then that can be metabolized and out of the system. But if that is blocked, definitely again there will be anabolism. Insulin is right act of function. Second, insulin also inhibits breakdown of triglyceride. Again, triglyceride ta বেড়ে যায়. Insulin breakdown ta করাচ্ছে না. Triglyceride, if triglyceride is broken down, ultimately it will again go into free fatty acid. That is broken, that is stop, again anabolic function. Last one, এটা ভুললে চলবে না. This is very, very important. Insulin stimulates malonyl-CoA, and malonyl-CoA is, যদি এটা পুরো মনে রাখতে না পারো, malonyl-CoA inhibits carnitine system. Okay, so carnitine acyl transferase যদি ভুলে যাও, এনজাইম গুলো মনে রাখা লাস্টের দিকে গিয়ে is very difficult. Try to understand one thing. Carnitine system, if that is on, that will increase the keto acid. That will increase the keto acid. ইনসুলিন কি করছে? সাপোজ কিটো অ্যাসিডের যে প্রিকারসর গুলো রয়েছে, তাদের we can deal with them in certain ways. কি ways রয়েছে? One way is put them into citric acid cycle. So once we put them into citric acid cycle, ওটা বেরিয়ে যাবে পুরোপুরি, or that will be broken down totally. But if suppose in our body keto acid level is very high, only citric acid cycle cannot handle that. So what will happen? Then it will move into malonyl-CoA pathway. Now malonyl-CoA pathway actually helps in reducing keto acid by inhibiting carnitine pathway. ম্যালোনিল কোয়ের ফাংশনটা কি? কিটো অ্যাসিড থেকে ফ্রি ফ্যাটি অ্যাসিড এর মধ্যে কনভার্সন গুলোকে করানো। ইনসুলিন থাকলে কি হচ্ছে? দ্য ম্যালোনিল কোয়ে ইজ হাই। ইনসুলিন যদি কম থাকে তাহলে দিস কিটো অ্যাসিড হ্যান্ডলিং উইল নট বি দেয়ার, which will lead to ketoacidosis. That's the reason why ketoacidosis happens in certain types of diabetes. যখন ইনসুলিন লেভেল কম থাকে তাহলে ইনসুলিনের ফাংশন হচ্ছে it acts on it, it stimulates malonyl CoA which reduces keto acid, free keto acid presence. Clear? Okay, so এইটা পুরোপুরি হয়ে গেলে we will move into the next topic. Done everybody, more or less? Okay. এটা this can come as a 10 marks question or একদম বড় কোশ্চেন হিসেবে আসতে পারে। whatever we have drawn এটা কি প্যারাগ্রাফে লিখে যাবে, 80% 90% স্কোরের জন্য সাফিসিয়েন্ট। তার বেশি আমি নিজে কোনোদিন পাইনি, আই কান্ট হেল্প ইউ, নিজেরা করে নিন। আচ্ছা, এবারে আসছে হচ্ছে ইনসুলিন রেগুলেশন। ইনসুলিন রেগুলেশন কি করে ইনসুলিনটা সিক্রেট হচ্ছে। সো strongest regulatory factor for insulin is blood glucose itself. blood glucose বাড়লে ইনসুলিন সিক্রিশনটা বেড়ে যাবে। এছাড়াও অনেকগুলো রেগুলেটরি ফ্যাক্টরস আছে। ফ্যাক্টরস গুলো ফার্স্টে লিখে নাও। দ্য ফলোইং ফ্যাক্টরস হেল্পস ইন ইনসুলিন রেগুলেশন। ফার্স্ট ইজ ইনক্রিজড ব্লাড গ্লুকোজ লিডিং টু ইনক্রিজড ইনসুলিন সিক্রিশন। পরপর এগুলো লিখে নাও। ইনক্রিজ এক নম্বর। দুই, এক্সারসাইজ। এক্সারসাইজ increases insulin secretion and increases insulin utilization. প্রত্যেকটা এক্সপ্লেন করব, জাস্ট পয়েন্ট গুলোকে ফার্স্টে নিজেদের খাতায়, তারপর তুমি আমাকে পয়েন্ট বলবে, আই উইল এক্সপ্লেন কাইন্ড অফ দ্যাট। সেকেন্ড ইজ ইনসুলিন এক্সারসাইজটা ইনসুলিন সিক্রিশনটাও বাড়ায় and insulin utilization-ও বাড়ায়। থার্ড ওয়ান ইজ sympathetic and parasympathetic stimulation, দুটোই বাড়ায়, sympathetic and parasympathetic stimulation. Fourth is GL CC increases insulin secretion. Fifth, which is a subset of this point, incretin effect. Incretin effect. Again, I will come to that. Few weeks back I have discussed this. Today in a separate light we will discuss this. Then blood amino acid level in blood, সেটা বাড়লেও ইনসুলিন বাড়বে, because কি there's a need for that amino acid to get into the cell. তো amino acid যদি ব্লাডে availability বেড়ে যায়, ইনসুলিন will come up and help push that amino acid into the cell. যেটা এক্ষুনি বললাম ফাংশনটা, ওইটা ওইভাবে হচ্ছে। ব্লাড FFA ফ্রি ফ্যাটি অ্যাসিড যদি বাড়ে তাহলে ইনসুলিন বাড়বে, ফ্রি ফ্যাটি অ্যাসিড যদি বাড়ে তাহলেও ইনসুলিন বাড়বে। ক্লিয়ার? কিভাবে পুরো জিনিসটা হচ্ছে। Whenever plasma glucose is crossing 110 mg/dl, beta cells are stimulated and because this happens in two stages. So this is known as this, then we will be moving into that. So this is known as biphasic insulin secretion. Insulin secretion. Time, the secretion will be this. That is the graph. Plasma insulin with time. এটা কখন? এই যে পিকটা নিচ্ছে, ফার্স্ট পিকটা নিচ্ছে, this is এই পয়েন্টটা হচ্ছে ব্লাড গ্লুকোজটা যখন শুট আপ, তার অর্থ থেকে এই পিকটা নিচ্ছে। সো ব্লাড গ্লুকোজ শট আপ করলে এই পিকটা বেসিক্যালি নিচ্ছে। হাউ থিংস আর হ্যাপেনিং, ওই দিকটা এই দিকের ছবিটাও এঁকে নেবে, উইল এক্সপ্লেন দ্যাট। তাহলে once again let us tie the whole thing. Insulin regulation, we are discussing after first part of the class, we have discussed the function of insulin. And regarding the function of insulin, we have discussed about insulin's action on carbohydrate. Its major target is to reduce glucose level in blood. That is achieved by two ways. One is either by storing glucose or by breaking down glucose itself. But it doesn't keep glucose in blood, first of all. Similarly, its major action is keeping the fat as it is and stopping the breakdown of fat. Even if fat is broken down, stopping it to go into the ketoacid pathway. And the third function is protein. So keep protein as much as possible. Whatever protein is already there in body, keep them as it is. Whatever amino acid is already there, push them into the cell so that they are not at least in the blood, at least in the cell. And once they are in the cell, help them to become protein. Kind of that. That's the whole pathway. প্রত্যেকটা পাথওয়ে যদি পরীক্ষাতে ভুলেও যাও, এত কিছু তো আছে, ভুলেও যাও, এই টার্গেটে লিখতে থাকবে। সো তুমি এই টার্গেটে ইউ উইল আন্ডারস্ট্যান্ড যে হ্যাঁ গ্লাইকোজেনোলাইসিসটাকে বন্ধ করতে হবে, গ্লাইকোজেনোসিসকে চালাতে হবে, নিওগ্লুকোজেনেসিসকে বন্ধ করতে হবে, লাইপোলাইসিসকে বন্ধ করতে হবে, লাইপোজেনেসিসকে স্টার্ট করাতে হবে। ইউ উইল আন্ডারস্ট্যান্ড দ্যাট। এই জিনিসটা থাকলে যে ব্যাক ক্যালকুলেশন ইউ ক্যান রাইট 4 পেজেস মিনিমাম। এত লেখো যাতে আমরা বিরক্ত হয়ে নাম্বার দিয়ে দিই। দ্যাটস দ্য ওয়ে, এইভাবেই তো হয় সবকিছু। কিন্তু যদি ভুল করে ওর মধ্যে উল্টো পাল্টা কিছু খুঁজে পাওয়া যায় তাহলে মুশকিল। বাট ইফ ইউ হ্যাভ এ লজিক্যাল কোরিলেশন, উইল ডেফিনেটলি বি গিভেন গুড মার্কস। তো লজিক্যাল কোরিলেশনটা এইখানে যে ইনসুলিনের মেইন টার্গেটটা কি, এইটি টার্গেট। এর মধ্যে নাও ইউ এরপরে ইফ ইউ ক্যান কিপ ইফ ইউ ক্যান mention name of the reactions, glycogenosis, glycogenolysis, kind of that, then you will be given good marks. তারপরে যদি এনজাইম লিখতে পারো তাহলে তো ভীষণ ভালো। এনজাইম মনে না থাকলে লিখতে গিয়ে পাশের লোকের থেকে ভুল লিখো না, তাহলে আবার চোখে লাগবে। সো এইভাবে স্টেপ, ফার্স্ট ইফ ইউ ক্যান রাইট, ইউ উইল বি গিভেন পাস মার্ক যদি তুমি প্রপার সাবস্ট্রেটটা কোন দিকে যাচ্ছে বলতে পারো। ইউ উইল বি গেটিং 70% অর অ্যারাউন্ড যদি তুমি লিখতে পারো যে কোন রিঅ্যাকশনটা এক্স্যাক্টলি হচ্ছে, যে এটা কিটো অ্যাসিড অক্সিডেশন হচ্ছে, ওটা গ্লাইকোলাইসিস হচ্ছে, এটাকে বন্ধ করছে, দ্যাট। এন্ড ইফ ইউ ক্যান অ্যাড অন দি এনজাইম, সো ডেফিনেটলি 90s ওইদিকেই যাবে জিনিসটা। ক্লিয়ার এভরিবডি? সো এইভাবে লজিকটাকে মাথায় রাখো, ইনসুলিনের লজিক। সো দ্যাট হ্যাজ বিন ডান ইন দ্য ফার্স্ট পার্ট। সেকেন্ড পার্ট অফ দি ক্লাস টুডে we are doing regulation of insulin. So how insulin is, insulin secretion, how that is controlled in body. এমনি তো আর হতে পারে না, there must be something. So first important thing is blood glucose. Blood glucose তার মানে কি, we are taking something through our mouth or maybe through intravenous, whatever, সেটাও তো blood glucose বাড়াবে। Whatever is happening, if blood glucose level is more than 110, insulin secretion will increase. From where? From beta cells. How? In a biphasic manner. Biphasic means initially insulin comes up, and as the pulse comes up, after a while that reduces, and then again it starts to grow. So prothome there is a sharp increase in insulin secretion followed by a gradual increase in insulin secretion. But now what is the reason? Reason is on this side of the board. So beta cell has grew where insulin itself, they are independent of insulin and they can sense glucose by themselves. Whenever glucose is more on that side, glucose will enter more. So this is again acting on the transmembrane glucose gradient. Just like hepatocyte, GLUT2 transmembrane glucose gradient এর উপরে কাজ করছে। And লিভারের ক্ষেত্রে ইনসুলিন কি করতো, ইনসাইড দি হেপাটোসাইট গ্লুকোকাইনেসকে ইনক্রিজ করে গ্লুকোজ লেভেলটাকে কমিয়ে দিত। এখানে কি হচ্ছে, এখানে ব্লাডেই গ্লুকোজ লেভেলটা উঠছে, সো ট্রান্সমেমব্রেন গ্রেডিয়েন্ট ইজ ইনক্রিজড, এন্ড দ্য মোমেন্ট ট্রান্সমেমব্রেন গ্রেডিয়েন্ট গ্লুকোজ এন্ট্রি, লেভেল অফ গ্লুকোজ এন্ট্রি ধরো খুব বেশি, ব্লাডে গ্লুকোজ প্রচুর, কন্টিনিউয়াস গ্লুকোজ ঢুকছে, যত ঢুকবে এখানে একটা ফিড ফরওয়ার্ড মেকানিজম হয়, যত ঢুকবে তত বেশি রিঅ্যাকশন হবে। ট্রাই টু আন্ডারস্ট্যান্ড, এন্ড দ্যাট রিঅ্যাকশন ইজ ফসফোরাইলেশন। কি ফসফোরাইলেশন হচ্ছে? দিস গ্লুকোজ ইজ phosphate. This glucose 6-phosphate conversion increases the ATP breakdown. Energy চাইছো তো increases the ATP breakdown. ATP, the moment more and more ATP breakdown is happening, more and more ATP movement is happening, this ATP বন্ধ হয়ে যাচ্ছে। So the thing is glucose entry, glucose conversion into glucose 6-phosphatase, increase ATP in the cell. This ATP is stopping ATP sensitive potassium channel to function. সেলের মধ্যে পটাশিয়াম বেশি থাকে না বাইরে পটাশিয়াম বেশি থাকে? Intracellular potassium concentration 140 plus, external potassium concentration 3, 4, 5. Clear? So এত গুণ 40 times 50 times রয়েছে। So ATP sensitive potassium channel, if that is closed, what will happen? Potassium exit will stop. Clear? ATP sensitive potassium channel open থাকলে কি হবে? Potassium will just go out. If ATP sensitive channels are stopped, potassium exit, if flux is stopped, what will happen now? Potassium level inside the cell will go up. So যদি potassium, is it a cation and ion? Cation. So যদি cation সেলের মধ্যে বেড়ে যায়, what will happen? Depolarization. যদি depolarization হয়, what will happen? Now this is a voltage sensitive, this is the voltage sensitive channel will respond to depolarization and it will open up. The moment it opens up, calcium from outside, and as calcium from outside comes in, intracellular calcium level goes up. As intracellular calcium level goes up, we already have insulin, preproinsulin, proinsulin, C-peptide, all these are already present in vesicles. So the moment calcium comes up, there will be exocytosis of stored insulin, exocytosis of stored insulin. So initial batch of insulin granules are open depending on rise in calcium by the process of exocytosis. This explains the initial peak. This explains the initial peak. Later peak, PG level, পরেরটা কেন হচ্ছে? তাহলে পিকটা নিল তারপরে কমে গেল because ready-made insulin is down now. Later one হচ্ছে secondary granules থাকে, হচ্ছে ভিতরে, and ultimately what cellular pH granules যেগুলো ইনসুলিন রয়েছে, but in the second batch they will open up and they will start releasing insulin. So that is the reason why slow progressive insulin rise happens after a while. So first one is the primary granules which are completely voltage sensitive, which are completely calcium dependent, and the second one is the secondary granules which are pH dependent. So as more and more glucose is has entered the cell, ultimately এগুলো তো pyruvatal লাইনে যাবে, so pH will come down. As pH will come down, the secondary granules will start firing. That's the whole thing over here. So biphasic insulin secretion, first is a voltage dependent process, second is a gradient pH dependent process. So first one is an electrical phenomena, the second one is a chemical phenomena. এতটা যদি নাও মনে রাখতে পারো, at least you need to understand this picture, where again I am just mentioning this picture, very very important. Also in pharmacology, particular drugs act on this pathway. So beta cells, their GLUT2 is present, which is independent of insulin, dependent on transmembrane glucose gradient. Whenever blood glucose is increased, transmembrane glucose gradient increases. As that increases, extra glucose gets into beta cell of pancreas through GLUT2 transporter. Inside the cell, after passing the GLUT2 transporter, glucose is phosphorylated, glucose 6 phosphate is formed, ATP production increases. As ATP production increases, ATP sensitive potassium channels are blocked. As ATP sensitive potassium channels are blocked, potassium efflux from high intracellular potassium concentration to outside stopped. So intracellular potassium concentration goes up, leading to depolarization. This depolarization opens up voltage gated calcium channel. Calcium enters into the cell. This calcium causes the exocytosis of the primary granules, which are almost ready insulin, which are just ready and sitting up over there. There is a secondary kind of a granules that helps in the secondary peak or secondary, plateau and going up, secondary rise of this, but that requires reduction of pH, and then only they comes up, and that happens if glucose continuously goes up. কেন এরকম বানিয়েছে? This kind of scenario, nature তো এমনি দিতে পারতো একটা প্রাইমারি সিঙ্গেল একটা ফেজ দিতে পারতো, নেচার এরকম কেন বানিয়েছে? সিঙ্গেল ফেজ দিলে প্রবলেম হতো যে গ্লুকোজ লোডটাকে দে কান্ট রেগুলেট। প্রথমে যেই মোমেন্টে গ্লুকোজটা যাচ্ছে বেরিয়ে যাচ্ছে ইনসুলিন। ইফ ইনসুলিন কিপস অন গোয়িং আপ, ইফ দেয়ার ওয়াজ নো চেক, আফটার আ ওয়াইল তুমি খাচ্ছ না তবু ইনসুলিন বাড়ছে, অ্যান্ড দেয়ার উইল বি হাইপোগ্লাইসেমিয়া। সো দ্যাটস দ্য রেগুলেটেড। তুমি যদি পারসিস্টেন্টলি গ্লুকোজ দিতে থাকো, only then this secondary granules will be activated, only then, and that is a slow process. Slow because you have to find out the gradual change in glucose level. So first glucose load যেই আসছে, that's a very quick process. Immediately, সেই জন্য first phase is electrical. Electrical is a quick process. Chemical is a slow process. আমি এটা বলবো না যে chemical is a slow process, তাহলে পৃথিবীতে বোম তার অস্তিত্বই থাকতো না। হ্যাঁ, chemical is not a slow process, but here at least এটাকে বায়োকেমিক্যাল গ্রেডিয়েন্টের মধ্যে রেখেছে। So that's why it's a slow process, slightly slower process. So persistent glucose যদি দিতে থাকো, only then you will be getting that phase. Clear everybody? জিনিসটা বোঝা গেল? এটা ছিল গ্লুকোজের ইনক্রেটিন এফেক্ট। এখানে যেটা the more exercise, the more GLUT4 expression, insulin dependent glucose transporter expression on the cell membrane level of cardiac muscle, skeletal muscle, adipose tissue. More and more glucose entry happens. So ঠিক আছে, incretin effect. I have discussed about incretin effect. Level is incretin effect doesn't require, or it is irrespective of your blood glucose, incretin effect acts. কিরকম ব্যাপারটা? তুমি কিছু খেলে, কিছু anything you have taken. Once you have, suppose you have taken one sweet, that sweet goes inside, and then that extra sweet will be absorbed. Once that sweet is absorbed, blood glucose level will go up. Understood? So once that blood glucose level goes up, only then this biphasic thing starts. Incretin effect is irrespective of blood glucose. What happens? Incretin effect depends on incretin effect is so it is independent of blood glucose level. It is just oral glucose can increase these hormones which will increase insulin secretion. That is incretin effect. কোশ্চেন আসে, আগেও একবার বলেছিলাম, কোশ্চেন যেটা আসে সেটা হচ্ছে ওরাল গ্লুকোজ ক্যান ইনক্রিজ অর ইনসুলিন লেভেল মোর দেন আইভি গ্লুকোজ। ওরাল গ্লুকোজ দিলে ইনসুলিন লেভেল বেড়ে যাচ্ছে, তার কারণ হচ্ছে দুটো রিজন। এক, ইনক্রেটিন এফেক্ট, প্লাস দ্যাট উইল বি অ্যাবজর্বড ইন ব্লাড। কিন্তু যখন আইভি গ্লুকোজ দিচ্ছি, দ্যাট ইজ অনলি ইনক্রিজিং দি ব্লাড গ্লুকোজ, ইনক্রেটিন এফেক্ট ইজ নট দেয়ার। Clear? So incretin effect হচ্ছে the reason why oral glucose is more efficient to increase blood insulin level than IV glucose. Very counterintuitive idea. আমরা কি ভাবি যে IV যদি কোনো কিছু দিই, action is more, and which is more or less in most of the cases that's the reason. That's what we see. But here it is a slightly different. কিছু খেলেই ইনক্রেটিন গুলো বাড়ে, সেটাই ইনসুলিনটাকে বাড়িয়ে দেয়। ক্লিয়ার? And these incretins are GLP-1, GIP, এগুলোর উপরে প্রচুর কাজ আছে, which in subsequent years you will come to know. Third one is the paracrine effect. Paracrine effect, what we have seen, glucagon by paracrine effect increases insulin release, and this insulin in a negative feedback way reduces glucagon. That's the paracrine effect over there. And paracrine means what? One type of endocrine cell having its effect in the vicinity of it. So alpha cell, beta cell, they are very close by. More and more glucagon. Glucagon বাড়লে কি হয়? Why this is given? কারণ কি গ্লুকাগনও যদি বাড়ে তাহলে সুগার লেভেলটা খুব আপ হয়ে যাবে। So they have been given as a paracrine level regulator. গ্লুকাগন বাড়লে ইনসুলিনটাকে বাড়িয়ে দিচ্ছে, and যখনই ইনসুলিন বাড়ছে it reduces glucagon. So সবটা মিলে homeostasis is maintained. So একটা পজিটিভ ওয়ে, নেগেটিভ ফিডব্যাক is running over there. So incretin is done, and this is actually a feed forward mechanism. এই যে প্যারাক্রিন এফেক্টটা, এটা হচ্ছে ফিড ফরওয়ার্ড মেকানিজম। ফিড ফরওয়ার্ড মানে যখন জিনিসটা চলতে থাকবে। হ্যাভ ইউ হার্ড অফ ফিড ফরওয়ার্ড মেকানিজম? হ্যাভ ইউ হার্ড অফ ফিডব্যাক মেকানিজম? পজিটিভ ফিডব্যাক, নেগেটিভ ফিডব্যাক তোমরা জানো। ফিড ফরওয়ার্ডটাকে সিম্পল একটা এক্সাম্পল দিচ্ছি। তুমি 4 মাস পড়াশোনা করোনি, পরীক্ষা আসছে, আজ থেকে পড়াশোনা শুরু করলে 1 মাস পরে পরীক্ষা। তুমি এই 2 দিন পরে ভাবলে বিরাট পড়েছি, কমিয়ে দিলে। 2 দিন নেটফ্লিক্স বেশি দেখলে, 2 দিন নেটফ্লিক্স দেখার পরে ভয় হলো, কিচ্ছু করিনি, বাড়িয়ে দিলে। আবার 2 দিন 3 দিন পর আবার সেই ওভার কনফিডেন্স চলে এলো, ঘুরে এলে একটু শান্তিনিকেতনে বেশিক্ষণ ঘুরে এলো। রাত 11টায় যখন লুকিয়ে পাঁচিল টপকে ফিরছো তখন আবার ভয় এলো। দুটো কারণ, ধরা পড়তে পারো, সেকেন্ড কিছু পড়া হয়নি, সব ভুলে যাচ্ছি মনে হচ্ছে। আবার দেখা গেল সেদিন অতিরিক্ত সারা রাত ধরে পড়লে, পরের দিন তার ফলে সারাদিন বিজি থাকছো, কিচ্ছু পড়া হলো না। দিস ইজ ফিড ফরওয়ার্ড। এভরি মোমেন্ট চেঞ্জ হচ্ছে, যতটা রিকোয়ারমেন্ট সেই অনুযায়ী চেঞ্জ হচ্ছে। ফিডব্যাক হচ্ছে প্রচুর পড়েছো তাই আর দরকার নেই, নেগেটিভ ফিডব্যাক। পজিটিভ ফিডব্যাক হচ্ছে প্রচুর পড়েছো কিন্তু নোবেল পেতে হবে তাই কন্টিনিউয়াস পড়ে যাও, দ্যাট ইজ পজিটিভ ফিডব্যাক। দিস আর দ্য টু ডিফারেন্ট। ফিড ফরওয়ার্ড হচ্ছে প্রতি মোমেন্টে চেঞ্জ, প্রতি মোমেন্টে। এটা সেরিবেলামে আছে, এটা এখানে আছে, অনেকগুলো জায়গাতে রয়েছে। ফিড ফরওয়ার্ড ইজ স্লাইটলি ডিফিকাল্ট টু ফ্যাথম এই মোমেন্টে, কিন্তু এইটা অপশন। এইভাবে জিনিসটা মানে প্রতি মোমেন্টে এইটুকু এগোলাম, এতে দেখে নিচ্ছি কতটা কন্টিনিউয়াসলি এফেক্ট এন্ড রেসপন্স, এফেক্ট এন্ড রেসপন্স এন্ড স্টিমুলাস, তার মধ্যে একটা কানেকশন। সো গ্লুকাগন একটু বাড়লে ইনসুলিনটাকে বাড়িয়ে গ্লুকাগনটাকে কমিয়ে দিচ্ছে। আবার ইনসুলিন বাড়াবাড়ি বেড়ে গেলে দেখা যাবে অন্য দিক থেকে গ্লুকাগন বেড়ে যাচ্ছে, কাইন্ড অফ দ্যাট। ক্লিয়ার? ইনসুলিন যদি বাড়াবাড়ি বেড়ে যায়, দেয়ার উইল বি হাইপোগ্লাইসিমিয়া। হাইপোগ্লাইসিমিয়া হলো স্ট্রেস রেসপন্স, কর্টিসল, গ্লুকাগন ওই দিক দিয়ে বেড়ে গেল। বেড়ে যেতে দেখা যাচ্ছে যে আমার স্ট্রেস রেসপন্স নরমাল হলো, কিন্তু দেখা যাচ্ছে সুগার প্রচন্ড হাই। এগেইন দিস গ্লুকাগন উইল বি শর্ট অফ ইনসুলিন, এগেইন থিংস উইল হ্যাপেন। সো একটু গিয়ে পুরো জিনিসটাকে চেক করে। সো দিস অল আর examples of very sensitive regulatory mechanism already present in our body by nature. Clear? So এইটা হচ্ছে প্যারাক্রিন এফেক্টটা ওই জন্য দেওয়া রয়েছে। প্রত্যেকটা লেভেলে, মলিকুলার লেভেলে they have given some kind of regulatory effect. কারণ প্যারাক্রিন থাকাটা কি হচ্ছে, লোকালাইজড ব্যাপারটা আমি বুঝতে পারছি। আদারওয়াইজ হোয়াট উইল হ্যাপেন, ইনসুলিন সেই ব্রেনে গিয়ে হাইপোথ্যালামাস স্টিমুলেট করবে, গোটা ব্লাড ঘুরে আসার পরে এভরি পাস, তারপর লিভারে গিয়ে চেঞ্জ করবে। ইট উইল টেক মোর টাইম। আমাদের গ্লুকোজ অনেক ফ্লাকচুয়েট করতো সেখানে, এখানে মোর অর লেস গ্লুকোজটাকে একই লেভেলে রেখে দিতে পারছে এই জাতীয় জিনিস থাকার জন্য। যেখানে যে ফ্যাক্টরিতে যেখানে ইনসুলিন তৈরি হচ্ছে, উই আর স্টপিং ইট ওভার দেয়ার অর উই আর ইনক্রিজিং দ্যাট। সো ইটস অল অ্যাবাউট দ্য ফ্যাক্টরি লেভেল প্রোডাকশন ইনক্রিজ অর প্রোডাকশন ডিক্রিজ। দ্যাটস প্যারাক্রিন এফেক্ট। আরেকটা হরমোন আছে, দ্যাট ইজ যে হরমোন সোমাটোস্ট্যাটিন। কিছু কিছু লোক থাকবে দেখবে তোমাকে সারাদিন বলে তোর দ্বারা কিছু হবে না, তুমি যাই করো তাও তোর দ্বারা কিছু হবে না। আমি তো অ্যানাটমিতে অনার্স পেয়ে তাতেও কিছু হবে না। হ্যাঁ, আমি তো এমবিবিএস এর সব সাবজেক্ট অনার্স, এমবিবিএস পড়েও কিছু হবে না। হ্যাঁ, আমি তো পিজি করেই বা কি হবে, কি অবস্থা ডাক্তারদের দেখছিস। হ্যাঁ, তারপর তুমি একদিন দেখা গেল ওই লোকটা মৃত্যুসজ্জায়, তুমি গিয়ে বাঁচিয়ে দিলে। বাঁচিয়ে দেওয়ার পর বলছো যে এবারে বলে এতেও কিছু হবে না, আমি তো এরকম লোক। কাইন্ড অফ দ্যাট। সোমাটোস্ট্যাটিন ইজ দ্যাট কাইন্ড অফ হরমোন। সোমাটোস্ট্যাটিন প্রত্যেককে বলে তোর দ্বারা কিছু হবে না। সোমাটোস্ট্যাটিন রিডিউসেস ইনসুলিন, সোমাটোস্ট্যাটিন রিডিউসেস জিএইচ, সোমাটোস্ট্যাটিন রিডিউসেস গ্লুকাগন, মানে ও গ্লুকাগনকেও নামিয়ে দেয়, ইনসুলিনকেও নামিয়ে দেয়। এরকম কিছু সোমাটোস্ট্যাটিন বন্ধু বান্ধব থাকলে জীবন অসাধারণ হয়ে যাবে। সোমাটোস্ট্যাটিন ডেল্টা সেল অফ প্যানক্রিয়াস এখান থেকে বেরোয়, বাট ইট হ্যাজ আ প্যারাক্রিন এফেক্ট অন অল অফ দেম। প্যানক্রিয়াটিক পলিপেপটাইডকে ডিক্রিজ করছে, ইনসুলিনকে ডিক্রিজ করছে, গ্লুকাগনকে ডিক্রিজ করছে, কখনো কখনো আমার মনে হয় ও নিজেকেও ডিক্রিজ করে, কাইন্ড অফ দ্যাট। তো সোমাটোস্ট্যাটিন সবাইকে থামিয়ে দিচ্ছে। তো দ্যাটস দ্য প্যারাক্রিন এফেক্ট। ক্লিয়ার? এইবারে নেক্সট থিং, আজকে জাস্ট স্টার্ট করে দেবো, এন্ড দ্য নেক্সট ক্লাস উই উইল মুভ অন টু ইট, দ্যাট ইজ ডায়াবেটিস। ইয়েস, মেজর জায়গা, এটা আগে স্টার্ট করবো। হোয়াট উই হ্যাভ ডান, এখান থেকে কোশ্চেন কি কি আসে। ইনসুলিন রিসেপ্টর, শর্ট নোট, আগের দিন করানো হয়েছে। বিটা সেল থেকে ইনসুলিন ইজ বিং সিক্রিটেড, এই যে ছবিটা। ফাংশন, মেটাবলিক অ্যাকশন অফ ইনসুলিন, সেইটা মেটাবলিক অ্যাকশন অফ ইনসুলিন গেল। রেগুলেশন অফ ইনসুলিন, দ্যাটস দ্য ফোর্থ ওয়ান। প্যারাক্রিন এফেক্ট বিটুইন সোমাটোস্ট্যাটিন, গ্লুকাগন, এটা চান্সটা কম, কোশ্চেন ফর্ম করা একটু ডিফিকাল্ট। ইনক্রেটিন এফেক্ট, শর্ট নোট। ক্লিয়ার? এই জায়গাগুলো হচ্ছে ভীষণ, মানে বাইফেজিক, এটা চাওয়া হবে এক্স্যাক্টলি কোশ্চেন সেভাবে কম করা হয়েছে, বাট বাইফেজিকটা দেখতে চাওয়া হবে, খাতায় লিখছো যে রেগুলেশনের মধ্যে। গ্লুট, শর্ট নোট, এসজিএলটি শর্ট নোট, গ্লুট শর্ট নোট, এসজিএলটি শর্ট নোট। ক্লিয়ার? আর যেগুলো তোমাদের বললাম, ইনসুলিন ইজ অ্যান অ্যানাবলিক হরমোন, শর্ট নোট অর এক্সপ্লেন ওয়াই। Insulin, after insulin taking, people complains of weight gain, explain why. IV glucose is less insulinotropic compared to oral glucose, short note or explain why. Incretin effect, এটাই গল্প, এটা short note or explain. Insulin or low insulin level leads to ketoacidosis, ওইখানে একটু আগে করেছি, short note or explain why. সিম্প্যাথেটিক প্যারাসিম্প্যাথেটিকের এক্সপ্লেনেশনের মধ্যে হচ্ছে এইটা বিটা সেলকে সিম্প্যাথেটিকটা সুইচ অন করে, সেটা একটা সেই কারণে বাড়ে। প্যারাসিম্প্যাথেটিকে যেটা হয়, তোমার ফুড ইনটেকটা বাড়ছে, সো ওভারঅল ওটা বেড়ে যাচ্ছে। প্যারাসিম্প্যাথেটিক ইজ সামথিং যখন ডাইজেশন ইনক্রিজেস, সো অল দ্য ডাইজেস্টিভ হরমোন ইনক্রিজেস, ডাইজেস্টিভ এনজাইম ইনক্রিজেস। ইন দ্যাট ওয়ে দিস ইনক্রেটিন এফেক্ট ইজ মোর স্টিমুলেটেড, ইন দ্যাট ওয়ে ইজ গ্লুকাগন মোর স্টিমুলেটেড, ইন দ্যাট ওয়ে ব্লাড গ্লুকোজ absorption more stimulated, so that is the way. Sympathetic কি করছে, স্ট্রেস রেসপন্স বাড়াচ্ছে, সো স্ট্রেস ইনডিউসড ইনসুলিন রিলিজ সেটাকে বাড়াচ্ছে। দুটোই বাড়াতে পারে, এন্ড ইট ইজ ডিপেন্ডিং অন দ্য সিচুয়েশন। খুব বেশি এটাই আলাদা করে কিছু বলার নেই, মানে দুটোই ডিপেন্ডেন্ট অন, দুটোই ইনসুলিন কিন্তু সিক্রিশনকে বাড়াতে পারে। ক্লিয়ার এভরিবডি? আরেকটা হচ্ছে যেমন স্ট্রেস যদি বাড়ে, গ্লুকাগন বাড়বে, কর্টিসল বাড়বে, সুগার বাড়বে, ব্লাড সুগার, ইনসুলিন বেড়ে যাবে। ক্লিয়ার? প্যারাসিম্প্যাথেটিক হচ্ছে তুমি খেতে পারবে বেশি, ডাইজেশন বাড়বে, অ্যাবজর্পশন বাড়বে, ইনসুলিন বেড়ে যাবে। সো ইটস অলওয়েজ আ ব্যালেন্স, দুটোই এক কাজ করে বলে মনে হয়, কিন্তু ইন আ ব্যালেন্স। তাতে আমাদের মেইন টার্গেট, বডিজ মেইন টার্গেট ইজ টু কিপ সুগার উইদিন 80 to 140 বাই এনি মিনস। ডায়াবেটিস করবো না, তার আগে একটা ছোট্ট জিনিস দিয়ে আজকে শেষ করব, ডায়াবেটিস পরের দিন করে দেবো। সো লেটস ডিসকাস সামথিং, এটা সকলে বলতে পারবে। সো নেম ওয়ান হরমোন দ্যাট রিডিউসেস ব্লাড গ্লুকোজ। Name one hormone that increases blood glucose. Glucagon. Name second hormone that increases blood glucose. Cortisol. Name third hormone that increases blood glucose. কি জোরে বলো, জোরে বলো। Epinephrine. Very good. Name another epinephrine. এক্ষুনি যেটা বলছিলাম, epinephrine sympathetic system. Name another hormone that increases blood glucose. Growth hormone. Growth hormone. Name another hormone that increases blood glucose. Growth hormone related রয়েছে। 10টা অবধি হরমোন আমি পাবো যারা ইনক্রিজ করে ব্লাড গ্লুকোজকে, হরমোন এন্ড দেয়ার তাদের মাঝামাঝি জিনিসগুলো। নেম সেকেন্ড হরমোন দ্যাট রিডিউসেস ব্লাড গ্লুকোজ। বলো, সেকেন্ড হরমোন দ্যাট রিডিউসেস, হ্যাঁ, ডিরেক্টলি no. So any direct hormone that reduces blood glucose. Insulin is a direct hormone that increases blood glucose. যেগুলো বললাম, cortisol direct hormone up, glucagon direct, GH direct, IGF direct, epinephrine direct. আরেকটা কিছু বলো জোরে, কি বলছে? মরফিন বাড়িয়ে দেয়, বাড়িয়ে দেয়। এপিনেফ্রিন, নরএপিনেফ্রিন, সেম লাইন। অ্যাড্রিনালিন নরঅ্যাড্রিনালিন, এটাই তো এপিনেফ্রিন নরএপিনেফ্রিন ব্যারিয়ার, সেকেন্ড হরমোন যেটা ডিরেক্টলি রিডিউস করে ব্লাড গ্লুকোজকে। ছেড়ে দিচ্ছি জিনিসটা, ছেড়ে দিলাম। এবারে চলে আসছি আরেকটা জিনিস, ইনিশিয়াল হিউম্যান পপুলেশন, হোমোসেপিয়েন্স, ইনিশিয়াল হিউম্যান পপুলেশন, এদের জীবিকা কি ছিল, অকুপেশনটা কি ছিল? হান্টিং। আর হান্টিং, গ্যাদারিং, তারপরে কি এলো? ফার্মিং, অনেক পরে এসে, ফার্মিং অনেক পরে এসে। তারপরে কি এলো? মোটামুটি অনেক পরে, মানে জাস্ট লাস্ট 200 years something has changed. If you think of something has changed, hunting, gathering, এই সেই করে করে ইন্ডাস্ট্রিয়ালাইজেশন, ইন্ডাস্ট্রিয়াল 200-300 years। তার আগে ইন্ডাস্ট্রিয়ালাইজেশনের কোনো চিহ্ন হয়তো ছিল, ইন্ডাস ভ্যালি সিভিলাইজেশন বা ডেফিনেটলি এই যে উইপন্স বানাচ্ছে এটসেট্রা, ইন্ডাস্ট্রিয়ালাইজেশন টু সাম এক্সটেন্ট, বাট উইপন্স কম লোক সবসময় বাড়াতো, আর উইপন্স এর ইউজ অনেক বেশি লোক করতো all the time. So basically what we see that majority of during the evolution আমাদের হান্টিং, গ্যাদারিং, ফার্মিং এই টাইপের জিনিসপত্রগুলোই ছিল। ও হান্ট করে আনছে খাবার, আমি ওকে হান্ট করে খাবারটা খেয়ে নিচ্ছি, দ্যাট ওয়াজ দ্য সিভিলাইজেশন। এটা আজও চলে, এখন এটার নাম হচ্ছে বিজনেস, এটাকে বিজনেস বলে। যাই হোক, তো এখন যেটা দাঁড়িয়েছে, ইন্ডাস্ট্রিয়ালাইজেশনের পরে দেয়ার হ্যাজ বিন আ ডেরিভেটিভ, মাল্টিপল ডেরিভেটিভ। লোকে হচ্ছে ইনসুলিন থেকে এটা কোথায় যাচ্ছে, পিপল আর থিঙ্কিং লাইক দ্যাট, যাচ্ছে কোন জায়গাতে আসছে। তো ইন্ডাস্ট্রিয়ালাইজেশনের পরে ইউ উইল সি যে অনেকগুলো সেডেন্টারি লাইফস্টাইল এটসেট্রা, অনেক জিনিস, এমন অনেক জীবিকা তৈরি হয়েছে, ইনক্লুডিং আওয়ার্স, এমন অনেক জীবিকা তৈরি হয়েছে। আগে যদি সকাল 10টার সময় মাই খাতে আমি আজ থেকে 2000 বছর আগে দাঁড়িয়ে থাকতাম, 5 দিন পর মরে যেতাম খেতে পেতে, কিন্তু এখন ইটস পসিবল, এখন স্টিল উই ক্যান সার্ভাইভ। তোমরা যে জাস্ট বসে আছো, এটা আদিম মানুষের কাছে এটা ক্রাইম। সকাল 10টার সময় এটা হরিণ না ধরতে গিয়ে এখানে বসে আছো, বসে কি সব শুনছো, ইনসুলিন বা আরে খেলে তবে তো পারবে ইনসুলিন, খাবারটা আনো, এরকম ব্যাপার। কিন্তু নাও দ্য থিং হ্যাজ চেঞ্জড ওভার লাস্ট 200-300 years, the thing has changed. এবং যেহেতু থিংস হ্যাভ চেঞ্জড, ইউ সি 100 থেকে 150 বছর আগেও আমেরিকাতে ডায়াবেটিস সেভাবে পাওয়া যেত না, যদি বা পাওয়া যেত দ্যাট ওয়াজ ইনসুলিন ডেফিশিয়েন্ট অর ইনসুলিন ডিপেন্ডেন্ট ডায়াবেটিস mellitus, which is genetic from birth. You can't do anything unless Banting and Best discovers insulin and starts giving this. 1921 সালে ইনসুলিন দিয়ে from dogs থেকে ইনসুলিনটা বের করে ইনসুলিন দিয়ে ডায়াবেটিক কিটোঅ্যাসিডোসিস এর পেশেন্টগুলোকে বাঁচানো শুরু হয়েছিল। তার আগে ডায়াবেটিস ওয়াজ আ লিথাল এন্টিটি। ডায়াবেটিস টাইপ 1, এদের ডায়াবেটিস হতো, দেন কিটোঅ্যাসিডোসিস, হাইপারগ্লাইসেমিয়া, ডেথ, কোমা, ডেথ, দ্যাট ওয়াজ দ্য অনলি থিং। ডায়াবেটিস ওয়াজ ইভেন ওয়ার্স দ্যান ক্যান্সার এট দ্যাট টাইম টিল 1921 এ ব্যান্টিং অ্যান্ড বেস্ট এবং তার মধ্যে একজন 3rd ইয়ার মেডিকেল স্টুডেন্ট থাকার সময় ডিসকভার দ্যাট। 4 জন মিলে কাজটা করেছিল, একজন স্ট্যাটিস্টিশিয়ান, একজন কেমিস্ট, একজন প্রফেসর, একজন 3rd ইয়ার মেডিকেল স্টুডেন্ট। ওই সময় ওরা এই জিনিসটাকে বের করেছিল, and they worked on dogs first and then from there it went on. So basically my point here is body has been given with 10 glucose up hormones and only 1 glucose down hormone. Clear? ইনসুলিন ছাড়া নেই, তাই জন্য তোমরা আনসার দিতে পারছিলে না। Why this? এই কারণ this is, এটারও একটা নাম আছে, we call it thrifty gene hypothesis. thrifty gene hypothesis টা হচ্ছে ইভলিউশনারি ওয়েতে নেচার জানে যে আমাদের খাবার কম জুটবে, ইভলিউশনারি ওয়েতে নেচার এটা জানে। ইট ইজ অনলি লাস্ট 100 years we are in somewhat food surplus condition, যে খাবার বেশি আছে, আমরা ভাবছি ও প্লেটের অর্ধেক ফেলে চলে যাওয়া যেতে পারে, ফাস্ট ফুড খাওয়া যেতে পারে। নেচার অর দ্য কন্ডিশন ওয়াজ নেভার লাইক দ্যাট। একটা হরিণ মারবে 3 দিন ওইটাই খাবে, তাতে যদি পরের দিন আবার পাও অনলি দেন ইউ ক্যান ইট, প্রেফারেবলি অর মানে আই মিন যদি তুমি না বাঘের পেটে চলে যাও। এই ছিল তখনকার সিনারিও। এখন এসে গেছে যে কেএফসি ভালো লাগছে না, একটু গোল ঘরে গিয়ে খেয়ে আসি। নেচার জানতো না এটা, নেচার এটা জানে না। দ্যাটস ওয়াই নেচার হ্যাজ গিভেন ইউ সো মেনি আপ রেগুলেটিং হরমোন এন্ড অনলি 1 ডাউন রেগুলেটিং হরমোন। এবং এই কারণে ডিউ টু চেঞ্জ ইন লাইফস্টাইল অল অফ আ সাডেন ইভলিউশন ডাজন্ট হ্যাপেন ইন 100 years, সো ইভলিউশন ইজ নট এবল টু ক্যাচ আপ উইথ ইট। এই জন্য এত টাইপ 2 ডায়াবেটিস বেড়ে গেছে। ক্লিয়ার এভরিবডি? দ্য অল অফ আ সাডেন সেডেন্টারি লাইফস্টাইল পুরো জিনিসটা চেঞ্জ করে দিয়েছে, এবং চেঞ্জ করাতে কি হচ্ছে, উই অনলি হ্যাভ 1 হরমোন দ্যাট উইল রিডিউস সুগার এন্ড 10 হরমোনস দ্যাট উইল গো আপ। আর প্রচুর রিজন, স্ট্রেসের রিজন বেড়ে গেছে। ফার্স্ট ক্রপ কত পাবো, পাস করবো তো পাশে কে বসবে ফার্স্ট প্রপে, যে বসবে সে আদৌ জানে তো? এই দিস থিংস আর কন্টিনিউয়াসলি রিলিজিং কর্টিসল, দিস আর ক্রনিক স্ট্রেস, দিস আর ক্রনিক স্ট্রেস। এটা বলে এলাম, এইটা দেখছিলাম, এটাই বলে গেলাম শেষ মোমেন্টে, দিস আর ইনক্রিজিং দ্যাট। এই কারণে এই প্রবলেমগুলো কন্টিনিউয়াসলি হচ্ছে। এই জায়গা থেকে উইল বি মুভিং টু ডায়াবেটিস নেক্সট উইক। হ্যাঁ, ডায়াবেটিস, এবং আমার মনে হচ্ছে আর দুটো ক্লাস উইল বি রিকোয়ারিং ইন এন্ডোক্রাইন প্যানক্রিয়াস। ওকে,
So we are finishing this developmental aspects of urinary system. In the previous lecture, we talked about the development of, just to have a quick brief overview, whatever we have learned about the kidney development, which is a kind of like complex formations of the development in the urinary system. Now in the kidney, we learned that kidney had three sets of the kidneys which is available. We call them as pronephros. Then we have this next version of the kidney, we call them as mesonephros, and then we have the final version, we call them as metanephros. Along with that, we had a tube, we call them as nephric tube. That nephric tube, we call them as mesonephric tube, which has been utilized in different forms of formations of the kidney. From that mesonephric tube, a outpouch growth, we call them as ureteric bud, which interacts with this intermediate mesoderm, which is a nephrogenic tissue, lies there and forms this particular adult versions of the kidney. Now these adult versions of the kidney, which is metanephric kidney, develops in the region, we call them a sacrum region. But finally we see the kidney is placed under the diaphragm, so it has to climb up, we call them as ascent of the kidney. So that ascent of the kidney is occupied or kind of like triggered by the space or the growth of this lower part of the abdomen, which is growing at the lower pace compared to the upper part of the abdomen, and in search of better blood flow, it moves to the upper part of this abdominal cavity. Now as this particular kidney moves up, it rotates, we call them as rotations of the kidney, which is rotating 90 degree medially, so that the hilum of the kidney, which is facing medially. Now we learn this particular upward movement of the kidney, which is going up, could be arrested if the lower poles of the both kidney are united. We call such kidney as horseshoe shaped kidney. So that particular horseshoe shaped kidney is a kind of important short note for us to prepare for the examinations. And along with that, we learn about this particular abnormalities of the kidney development in forms of maybe have agenesis of the kidney, we may have a duplications of the kidney partially formed, we might have some kind of error in interactions in the formations of the kidney in forms of like ureteric bud and nephrogenic tissue not interacting properly can give rise to this multiple cyst formation. We call them as polycystic kidney. That also is a very important landmark question. We see that coming. Now from that part, we are finishing to the other end of the urinary system development, which is going over here is urinary bladder and the urethra and ureter, which is coming to develop from the sources which is coming over there. We'll focus on the development of the urinary bladder, which is a kind of like important source. Involving this urinary bladder, we see the clinical questions are asked. Two forms of these questions are asked in urinary bladder development part, mostly clinical, explain why kind of questions. So one of the questions which is involving this development of the urinary bladder, we have seen teaching in this supply exam, so which is that we are going to discuss. And another form of this urinary bladder developmental area, which is a complex form, we see that as well coming as a short note, which is coming out here. So for this part of the development, we have to go to this particular developmental part of the GI system. Now many times I refer to this particular entity about developmental area of the GI system, about the blind end of this foregut tube, blind gate of the gut tube, which is known as cloaca, which I insist on. It's a very important short note. There's a reason for it. So cloaca, which is a blind end of this particular pouch, which is getting over here, the gut tube. This part we call them as cloaca. So this particular cloaca, we've seen that is split into two parts by this developing mesodermal tissue. This mesodermal tissue, we call them as urorectal septum. The septum splits the cloaca into two parts, anteriorly and posteriorly. posterior part we've seen that developing into the rectum anal canal upper part, which later on united with the lower part of this region called as proctoderm forms this anal canal. And if that particular in between region which makes a membrane there, which is known as anal membrane, fails to rupture, we have a disease we call them as imperforate anus. And again, this is a very high yield exam short note question. Now coming to this particular cloaca again, so it's a kind of blind pouch which you seeing here running on this particular lower end of this caudal end of this fetus, and this particular cloaca is split into two segments, which is like a primitive rectum posteriorly, and anteriorly it is splitting this part, we call them as urogenital sinus. Now our class on this development of the urinary bladder will be involving over here on this urogenital sinus. So this particular cloaca, as you are seeing here, is split by this particular mesodermal tissue into two parts. Into this particular front part we call them as urogenital sinus, and the back side of the cloaca which is making this anal canal. So we focus on these front or ventral aspects of the derivative of this cloaca, we call them as urogenital sinus. Now this particular urogenital sinus has a channel, has a communication with this umbilicus. We call this communication as allantois. Now this communication, that is a tube which was there, which is known as allantois, is very important to note out here. So this such kind of communication remained in the gut as a diverticulum. If it remains, we call them as Meckel's diverticulum, which is like yolk sac communication, similar kind of thing. So in case of this, which was there in the midgut region. So in case of this particular lower gut, which is like a hind gut, has this particular urogenital sinus, which has a natural communication over here, which is known as allantois, which is communicated with this anterior wall of this embryo or anterior abdominal wall at the umbilical region. So this is what we have to know. Now before we go to this detail of this, so this is how we see that cloaca splitting into two parts, back side forming the rectum and front side forming the urogenital sinus. Now this particular urogenital sinus, as you can see, it having two parts, upper part which is little bit dilated, we call this dilated part as vesico-urethral canal, and the lower part we call them as definitive urogenital sinus. We do not have to remember this detail of the names, but we have to know this urogenital sinus, whatever you are seeing it here, this urogenital sinus has an upper part and a lower part. Upper part has a name and lower part has a name as well. So this particular upper part of this urogenital sinus, we call them as vesico-urethral canal, grows, enlarges and becomes this urinary bladder. Now this particular urinary bladder, as you see in the upper part, has a communication over here, which is we have mentioned here is allantois, A-L-L-A-N-T-I-O-S, allantois. So the communication channel matters to us. We'll be coming here later on when we're going to discuss about this particular clinical aspects of this particular bladder formation. This will matter to us. So this is, which is like a little bit having a kind of like a extra images which is showing us what is the allantois mean to. So this is the upper part of this particular urogenital sinus, which is known as vesico-urethral canal, which eventually forms the urinary bladder. On this particular, this region has a channel, has a communication, we call them as allantois, which is communicated with this umbilical region. Now this particular allantois normally is attached to the bladder apex, and this particular allantois goes into transformation with this embryonic development and becomes cord-like structure. Then we call this allantois as urachus. So this allantois makes into with developmentally urachus. So which is a cord-like structure which he is making out here. Now this particular urachus, when we are grown or getting into it, it's forming a ligament. We call them as median umbilical ligament. So this transformation of allantois we have to know. We'll be coming into why should we be knowing. So allantois is a kind of diverticulum, a channel connected to this apex of the bladder to the umbilicus, which with time progresses into cord-like structure. We call them as urachus. Then in adult, normally it becomes a ligament. We call them as median umbilical ligament, which is attached to the apex of the bladder. That's a key thing to identify the bladder apex. If you fail to identify the median umbilical ligament in urinary bladder, you cannot hold the organ in normal anatomical position. So that's what we have. These particular entities are important. And these things will be coming in exam vivas. So they will be asking you, whenever you're holding this organ, your urinary bladder as an organ, they will be holding it in anatomical position and they will be asking what helps to identify the apex. So you will be catching up this medial umbilical ligament and you will be saying this is medial umbilical ligament. They will be saying that, well, this is a medial umbilical ligament. What is the embryonic developmental source of medial umbilical ligament, or where from this medial umbilical ligament has reached to this particular part? So you have to come back to this particular urachus and allantois and then on the clinical things, slowly. So this is a part of this particular development. So urinary bladder, if it is, you are seeing here, which is making over here, majority of the urinary bladder along with this particular medial umbilical ligament develops from the urogenital sinus, which is forming this region and along with this. Now, this is not all. There is another inclusion which is a kind of important part about this particular urinary bladder developmental part. So we see that this developmentally, when this particular kidney system develops, we find a tube existing on this region. We call them as nephric duct. So that nephric duct, in case of the mesonephric kidney, we call them as mesonephric duct. So this particular mesonephric duct, we see that in the purple shadows we have highlighted over there. So this particular mesonephric duct on the right side and the left side, you see there is incorporated within this vesicourethral canal. So as the time progresses, a part of this ureteric bud, which is within this particular vesicourethral canal, slowly gets absorbed. And eventually, this particular absorbed right side and left side mesonephric duct, D, E and F, see that united together as the kidney moves up, this mesonephric duct pulled up and comes closer and united, and partly it is absorbed and forms the triangular area on a bladder. So in a bladder, if you open it up, we have a trigonal area, we call them as trigon of the bladder. So that particular trigon region of the urinary bladder is sourced or development coming from the united caudal end of the mesonephric duct. So the bladder, whenever you are addressing the union or formations of that, it has two sources, urogenital sinus, upper part of it, and the trigon part of the bladder is formed by union or absorption of the caudal part of the mesonephric duct united from the trigon of the bladder. Now this particular mesonephric duct is sourced from the mesodermal tissue. So this region, which is a trigon of the bladder, the epithelium over there is sourced from mesodermal origin. So whenever you are getting this particular bladder, most two common questions we ask about is the apex of the bladder and the median umbilical ligament. That's one thing. Again, interior to the bladder, most remarkable feature, we ask about trigon of the bladder and of course to the source of the trigon of the bladder. The source of the trigon of bladder is coming from union of this mesonephric duct, right side, left side, caudal, united, and that's why this is sourced as a mesonephric duct, which is forming the trigon of the bladder. And trigon of the bladder has a boundary which you know that is the lower part is formed by the urethra or opening of the urethra, and on the side is trigonal area. Now this trigonal area of the urinary bladder is formed by the absorbed caudal end of mesonephric duct. So this is the two informations regard to the bladder development we shout for whenever you are given in an exam questions. All right. So now coming to this particular end summary part, which makes these things easy. So the bladder, which is sourced coming from different regions. So we summarize the bladder as a development is concerned. So lining epithelium of the urinary bladder is endodermal, that is coming from the urogenital sinus. So everything lining epithelium, that means the cells, transitional epithelium, is present within this urinary bladder is coming from this vesicourethral part, which is the urogenital sinus, endodermal, that means the cloaca, where from it is coming eventually, except this trigon region, which is coming from this mesonephric duct, which is a mesodermal in origin. So this difference you have to keep in mind whenever is coming for this bladder, or if you are given this organ to hold or any question related to it, these two areas will be highlighting. The source of the trigon, where from it is sourced, and rest of this particular epithelium source. These are the two entities. Next is the muscle which is present in this connective tissue muscles which is present within this bladder is of course this mesodermal. So muscle will be coming from the splanchnoporous mesoderm, which is coming from the lateral plate mesoderm, and urachus of course we mentioned about, which is transformed in adult median umbilical ligament, and so on so on. So this is in short about bladder development, not much of complex thing. Once again, so this particular bladder develops from this particular cloaca. So as a result of this, cloaca comes as a very important short note. Whenever this particular element is given, this cloaca you have to write it down, which is having this particular complex thing which is coming. Partly it's contributing to this particular GI system, partly it is contributing to the renal system. Which part is which part and what are the transformations you are coming here. And in case of the bladder development, this particular urogenital sinus, which is having this particular upper part, which is known as vesicourethral canal, which is the upper part. So if you see this particular vesicourethral canal, which is this dilated part of it, it's forming this urinary bladder. And this particular trigon region is absorbed from this caudal end of the mesonephric duct on the right and left side, which is united from this trigon of the bladder. So this is about the bladder development, and along with that, the bladder region, which is the urogenital sinus, has a natural outflow region, which is known as the allantois, which normally should be transformed into cord-like structure and ligament subsequently with the time, and this particular entity is an important landmark. Now from this region, this is the end or in nutshell, the togetherness of the bladder development of the different sources of the bladder, which is kind of like a very important for your exams as MCQs or any other exams. Now coming to here, the clinical problem related to the bladder. So this is what we see that coming in a supply exam this time. So dribbling of urine or smell of the urine is coming through umbilicus in a newborn. So this is an exam question, you have to explain it why. And newborn, which is just born kid, which is having a kind of smell of the urine present in this umbilical region, or you see that once the baby crying, urine is coming out through the umbilicus. So similar kind of thing we talked about before in the GI system development, passage of stool in case of the umbilicus, passage of stool coming from the vaginal tract, passing of stool coming through the urinary system, etc., etc. Those are the things we talked about beforehand. So this is again dribbling of the urine, which is coming through umbilicus. So there must have some connection or some channel with this urinary bladder with the umbilical region or anti-abdominal wall. So which is, for that we have to go back to the developmental part, which is coming because of this entity. We call them as the allantois or persistent of the allantois. In forms of this we call them as urachus. So this particular urachus, which normally should be median umbilical ligament, is not formed medial umbilical ligament, rather it keeps this particular tract patent. So as a result, it has a communication with the bladder with this particular anterior abdominal wall through the umbilicus. So when the baby cries, that means when you have the increased intra-abdominal pressure, with this region, it squeezes out some urine through this particular fistular tract, through the urachus, and escaping through this umbilicus. You know, if this tract is very well patent, it will be dribbling the urine, and if it is less formed tract existent, that means the smell of the ammonia or smell of urine will be escaping through here, and it will be smelling umbilicus region with this urine smell. So this is about persistence, complete persistence with the sinus tract. We call them as urachal fistula or urachus tract, which is present over here, which is not been transformed into median umbilical ligament in case of this newborn individual. This is one entity. Second thing, it could happen. See this extreme. So this particular urachus, you see that partly it formed with this median umbilical ligament, partly it has a tract there and it has a sinus existing. So this part of this urethus, which is close to this anti-abdominal wall, is having a tract. Now, if this tract is there, we have some tissues in it. And this particular tissue, if it is infected, that means we have an infection which leads to formation of the pus. So, this kind of individual will be coming to your clinic, maybe adult, maybe young individuals, not necessarily it had to be kids, coming with these recurrent infections over here, pain in the abdomen, in the umbilical region, and coming with the discharge of pus through umbilicus. So, it happens over here with course of the time because of this persistence of the urethral sinus, which is a blind sinus which is making out here, making a kind of recurrent point for infection, because blind pouch, if it is there, it's prone to have infections. So, this particular urethral sinus, which is partly formed, partly absorbed by this medial umbilical ligament, can lead to formations of the infections and abscess formation subsequently. There's a third entity could be there, which is known as urethral cyst. So, this part, distal part and the proximal part is absorbed, the central part is unabsorbed and remains and holds a tissue along with some kind of fluid in it, can form a cyst-like structure. So, inside the abdomen, if you just like to see that, they might have some kind of nodule present, or maybe an incidental finding doing an ultrasound on the abdomen, you find this cyst existing and it is maybe mobile or maybe they are tense. Now, this particular cyst, if it is infected once again, can burst open. Then again, it has another complication. So, this particular urethral cyst also a possibility if you have a partially patent urethus present in here. So, if it is completely present, partially present, partly absorbed, partly non-absorbed, depending on which part is absorbed, we have different disease process. These all three sets of things are exam question. Along with that, when you move to the clinics, we find the disease which is having this region which is coming over this urethral fistula, which is present around the umbilicus only. And that particular skin over there is having kind of infections and eroded, can lead to formation of a tumor. We call them as raspberry tumor. So, that kind of tumor formation can also happen in this kind of persistent urethral sinus or urethral cysts. So, these are your exam question. We see that commonly hitting in examinations. Explain why kind of question, why this things is happening, and explain with a diagram and proper embryological explanation with it. So, this is one set of things we see that. Another variety, which is a grave variety of a developmental error, we call them as ectopia vesicae. Now, what is this? So, we have a kid born like this, which is like opening of the infraumbilical part of the abdomen. So, the kid which are born who is having this particular lower part of the abdomen is open, and you see that this protrusion of the urinary bladder, this tiny thing we are seeing here, the urinary bladder is coming out. So, what you are seeing here, this is not a formed urinary bladder. That means the apex and this part is not formed. What you are seeing here is the posterior wall of the urinary bladder. Now, this particular individual who are coming with this deficient anti-abdominal wall with this dribbling of the urine through umbilicus or through anti-abdominal wall. So, this individual do not have a formed anterior infra-umbilical portions of the wall. So, below the umbilicus, the abdominal wall is not formed, leading to this particular urinary bladder popped out outside, and the anterior wall of the urinary bladder is even not formed. So, the urine, whatever is produced through this kidney is coming out and continuously it is dribbling out. So, this is an individual case, it is a kind of grave entity which see that happening where we have this particular disease, we call them as ectopia vesicae, where this urinary bladder is expelled outside. It may be associated with other types of urinary bladder error, like maybe have kind of penile problem if it is a male individual. It may have some kind of other birth defect as well with the joint problem, etc., etc. It's not a very uncommon, but not a very common disease as well. It happens in a incidence of 1 in 10,000 live births. So, we'll see that urinary bladder is completely out. Now, this is what you are seeing here. Now, what makes this particular development error to find? So, this lateral folding, we see that happening with this particular embryonic folding, tail fold, cranial fold, which is happening, and as well as lateral folding which is happening. When it is the lateral fold which is happening in the lower part of the abdomen, it is happening as a lower pace. So, where the rest of the organ is formed, this particular pacing of this folding of the lower part of the abdomen, which is lateral folding, is slowly and sluggishly happening. That leads to this particular bladder which is trapped in between this closing part of the lateral folding. So, when this particular skin meets with this particular mucosa of the urinary bladder, it stops. It gets the signal, we have to stop. But still, it is yet to close this anti-abdominal wall. So, that is why we have this particular phenomenon forming here with embryological basis, incomplete formations or slow formation of this lateral folding of the embryo, and leading to this failure of this muscle tissue to migrate to this anterior part of the abdominal wall, and leading to this particular defect, which is infra-umbilical part of this anterior abdominal wall deficiently formed, leading to the protrusion of the bladder, which leads to this opening of the bladder to the exterior and leaking of the urine through anterior abdominal wall. So, these are the two entities we have to look for. So, see that what clinical case is given to this particular clinical problem. If it is given is the infra-umbilical part is deficient or not, that the entity, then you have to just like give this particular perfect explanation, or you see that everything is formed in this individual, only this particular urachus which is present. So, in this individual which is having this particular patent urachus or allantois, persistence of allantois, that is also like the same thing is there, where this anti-abdominal wall is completely formed. We do not have any deficiency in the anti-abdominal wall in this particular individual, where we have this particular anterior wall deficiency in this ectopia of the size. So, these are the two important areas. There are several other bladder anomalies are there. We are highlighting these two only because we see this frequently coming in an exam, explained why kind of questions. This is what we have this in a urinary bladder. Now, rest is very short about the rest part of the development. So, we talk about this particular ureter. Again, this is a very easy thing about this ureter. We just do not want to dig deep about this ureter. So, ureter develops from this particular ureteric bud. So, ureteric bud, which you see that coming from the lower end of this mesonephric duct, grows upwards, mixed with nephrogenetic tissue, mix and interactions, and forms this particular adult kidney, which is known as metanephric kidney. So, this particular ureteric bud, which is growing between this particular region from this which is lying between the renal pelvis and this vesico-urethral canal, in between that forms a ureter. Now, there are errors of this particular ureter development as well. So, for example, in case of this ureteric bud, one should be on the right side, one should be on the left side. So, this particular, instead of this particular ureteric bud, which is formed by the one on each side, may have some anomalies when it is developing. It may have not developed on a one side, other side is developing, and as well at the same time, it may have two of them developed, or partially it has formed and partially may not have formed. Based on this particular entity, we see this particular ureter having some kind of abnormality. You see that we call them as duplication of ureter, where you have two ureter. So, this particular ureter is present, duplications which is present to maybe there in a one side, other side may be normal, or maybe both sides have got this kind of duplication. This duplications of the ureter is maybe partial or maybe complete. That means, it may be present entirely throughout length and it is ending in a different opening urinary bladder, or may have this coming as two of them and then joining meanwhile and then making a single and joining like this. So, this particular kind of like a error in kind of like a ureter or formations of the ureter happens if you have any error on the ureteric bud development. And where from this ureteric bud develops? It develops from this guy, mesonephric duct, which is going to end to this particular urinary bladder, and this particular mesonephric duct lower part gives a out pouch which is known as ureteric bud, which is growing. So, if this particular ureteric bud is 2, it may give rise to 2 ureter, or maybe just like a splitting in between may give a partial duplication or complete duplications. So, this is the two entities about the ureter development. Other thing, this particular ureter have got muscles in it. So, this particular ureter which is developing may have a kind of hypertrophy of this particular muscle, which is leading to this particular dilatations, or may have some kind of obstructions in between when it is developing. So this can lead to dilatations of the ureter. We call them as hydroureter. It may be associated with the disease of the kidney or disease of the urinary bladder. So when you have this particular disease on this region, stone or something, it is pushing this particular urine throughout here and making an extra pressure, letting this particular ureter dilate. Same thing, you may have the obstructions at this particular opening point over here when this ureter is coming here. So we have an increased pressure. So urine is not normally coming through. So ureter is contracting, leading to the dilatations of ureter. Maybe unilateral dilatation, maybe bilateral dilatations. It could be there, depending on whatever the things. Now, this particular ureteric dilatations and errors, which is quite common when you see in a clinical life. But in an exam purpose, on an embryological basis, where you see that this question is not very frequently tested in examination. Rather, we see more commonly the bladder error or bladder defects, which is more commonly is coming as an exam question. Now, last not by the list, which is formations of the urethra. Now, we see that urethra in case of the male has a longer course, in case of the female it has a shorter course. So, this particular urethra is divided into few parts like prostatic part, bulbar part, and then membranous part, membranous part, then bulbar part, penile part. In case of the male individual, sequentially. In case of the female, it is like after the bladder, it is like the urethra, which is present in the deep pouch, which is like the membranous urethra, and then it is going to the exterior, which is short in length. So, in case of the male, the urethra develops from this particular region, we call them as urogenital sinus. So, again coming back to the urogenital sinus, the lower part of the urogenital sinus, we call them as pelvic part. So, this particular urogenital sinus, which you begin with in the cloaca formation, this urogenital sinus, which is having a narrow tapered part. So, we divide this particular urogenital sinus into two parts. Upper part dilated, we call them as vesicourethral canal, which forms this urinary bladder. Now, this particular urinary bladder coming from this upper part, which is developing from here. The lower part, we call them as the pelvic part, and then for the lower part, we call them as phallic part. So, this lower part of this urogenital sinus, we call them as pelvic part, and then on, we call them as phallic part. So, from this pelvic part of this urogenital sinus, the part of the urethra developing here is the prostatic part and the membranous part or the deep pouch part, which is developing in here. And this phallic part develops from the stimulation coming within this particular genital tubercle. So, when we are going to talk about this particular genital part of the development, we will be coming in. So, the source of the urethra is from this particular same urogenital sinus, the pelvic part, which forming the prostatic urethra and membranous urethra that is present in the deep pouch. After that, part of the main urethra, which is like a bulbar or penile part of the urethra, develops from the part of the urogenital sinus, which is known as phallic part, develops into it. Now, this development of the phallic part, it develops along with this male external genital development, which develops from a source, we call them as genital tubercle. So, genital tubercle is formed from the male external genitalia developing, which is stimulating this phallic part to be a part of the developmental source and gradually incorporated within it. So, this is how this urethra in male develops. In case of female, it is plain and simple. It develops from the pelvic part of the urogenital sinus. That's it. So, to summarize, whatever you will be emphasizing in this kidney system development, you will be emphasizing on development of the kidney in terms of this movement of the kidney. We call them as ascent of the kidney. You will be preparing that very well. So, that is very important high in exam notes. You will be preparing one clinical explanatory kind of things is known as the horseshoe shaped kidney, or why this particular horseshoe shaped kidney, the upward movement or ascent of horseshoe shaped kidney is arrested by inferior mesenteric vessels. This is one kind of explain why kind of question we frequently see in examination. Other thing, we see that polycystic kidney, we call them as congenital polycystic kidney. So, what are the embryological bases of formations of the polycystic kidney, which is known as congenital polycystic kidney, which is very important for us to develop. Then coming to the bladder, these things which is we talked about here, escape of the urine or in a smell of urine coming from umbilicus or in a cyst or a kind of sinus which is present to this particular umbilicus, which is leading to these particular clinical things, which is explained why. Other thing, ectopia vesici, which is a kind of exam embryological explanatory kind of areas which you have to prepare regard to this urinary system on here. All right, that's it. Thank you.
I think you have not been covered the thyroid gland in anatomy, isn't it? In the last two batches, I found that. I mean, so just you from your class 12 knowledge, you will be able to know the thyroid gland. At least you have that some amount of idea about the thyroid gland. It is located in front of the neck. It is having two lobes, and it is connected by isthmus. And its average weight is about 25 grams, but it varies widely from person to person. At the end of the lecture, I will show you some still photograph of enlarged thyroid and also video how you can see it. Now, if you make an histological section of the thyroid gland, you will find a structure like this, a follicular structure of thyroid, in which at the central part there is a colloid in which thyroglobulin molecule is available. I mean, where the thyroid hormone is synthesized. Normally what happens, we take iodine from the diet, that iodine is absorbed by the intestine and then it is circulated, transported through the blood into the thyroid gland where it is taken up. You can say that this is the area, this I am going to tell you step by step in detail. So this is this one cell. I am now concentrating on this cell itself. This cell, that is the basolateral surface, that is the capillary surface, from where the blood iodine is taken up by sodium iodine symporter and it is transported and then it is incorporated, this iodine is incorporated in the thyroglobulin molecule and then monoiodotyrosine, diiodotyrosine is formed and then it is T3, T4 is also formed and it is released and finally in response to TSH, this iodine is taken up and also T3, T4 is released. I will show this thing step by step. The important relevant information, I would say that daily requirement of iodine in adults is basically 150 microgram on an average. But for just like anything, this requirement is also increases in pregnancy. It is present in the drinking water. Its high amount is present in sea fish and sea salt. Smaller amount is present in milk, meat, vegetables and cereals. There is no need to write. Only thing is that please read this thing. Whatever is written in your book, you read it. But these are some of the information which I am sharing is for your own information. Now 90% of the iodine comes from food and rest from drinking water. So basically from food we get iodine. Now iodine content of the soil is the most important determinant of the iodine content in the water in that particular geographic area. And because of the water, the locally grown foods and animals, we subsist on basically fish, meat, the animal graze on that fields, so the iodine gets also incorporated in those animals and we get from dietary sources iodine. So iodine deficiency in area is basically geochemical in nature. Well, where the geochemically that area is iodine deficient, then only you will get an iodine deficiency. An adult human body contains 25 to 30 milligram of iodine and 80% of total iodine is stored in the thyroid. Blood level is 8 to 12 microgram per deciliter, which will interpret. Earlier, before the advent of T3, T4, TSH, protein-bound iodine used to be an investigation which was rarely done in a very few centers. That is now an historical test from mid-80s. Now, because of the iodine deficiency, iodization of salt is recommended. Whatever salt we collect, whatever salt we take is basically iodized salt. And if you look at the content, that Tata salt, just have given every salt is will give an. See, this is not an advertisement. This is because you all have this food. This is available in the market, and we purchase it. Many persons purchase it, and I want to say that iodized salt gives total iodine. Okay. Try to know the figure here. This 2 milligram of iodine is present. per 100 grams of salt. So that is 2000 microgram per 100 microgram. Okay, 1 gram contains 20 microgram. WHO recommendation of salt intake less than 5. Usually we take 3 to 4 grams of salt per day. So from the salt itself we get 60 to 80 milligram of iodine. So ideally there should not be any iodine deficiency. So thyroid hormones in our population cannot be due to iodine deficiency because we are consuming iodized salt. That was not there. If you go back to before 80s, we used to take normal salt which was not iodized. So formation of that iodine now we take, and this is taken up from the blood and concentrated by the thyroid. This step is stimulated by TSH. You see at the basolateral level, the basolateral membrane, there is a TSH receptor, and this step is stimulated by the TSH. TSH comes from pituitary and it binds to the TSH receptor and it activates, and because of that activation, this iodine is taken up by the thyroid acinar cell and it is incorporated, and this is inhibited by thiocyanate and perchlorate. The second important step is that you must remember this thing, that uptaken iodine is oxidized to active iodine and iodine is converted to elemental iodine by thyroid peroxidase enzyme. Thyroid is the only organ by which this oxidation takes place. Please try to remember, if the patient suffers from hyperthyroidism, we need to give drugs, and that drug inhibits some biochemical step. The anti-thyroid drug inhibits this step. If this step is inhibited, then thyroid hormone iodine cannot be incorporated into that thyroglobulin molecule. Consequently, no T3, T4 will be formed. This is basically iodination. I will tell you that you just remember this important step, that thyroglobulin molecules, there are several amino tyrosine residues, but out of that usually 35 tyrosine residues is iodinated on the intact molecule. This is tyrosine molecule, and if a single iodine atom is attached, it is called monoiodotyrosine. And if two atoms are iodide attached, it is called diiodotyrosine. Now, if one MIT and if one DIT is attached, that is called T3, or triiodothyronine. 3 iodine atoms are there, you can see here. And if 2 DIT is formed, combined together, then that is called T4. Okay, that is thyroxine. This is it is given in the form of drugs when the patient suffer from hypothyroidism. This you must remember. Not we don't give a T3. So this coupling, as I told you, the two molecules of diiodotyrosine forms 4 iodide atoms, and 99% of the T4 is formed by the thyroid gland. This 99% figure is I've taken from D.M. Vasudevan's book. Triiodothyronine means one MIT and one DIT. You must tell this thing properly during your examination. Now remember, thyroxine is produced, that is T4 is produced 100% by the thyroid gland, but T3 is produced peripherally. 80% of T3 is produced peripherally from deiodination of T4, and only 20% of T3 is secreted by the thyroid gland. Now, this iodination and coupling takes place in the follicular stage, which I have said you, and this is under the control of pituitary by TSH through hypothalamus. This regulation I will tell you, and blood levels of T3 and T3. These I will emphasize a little later. The fourth step is storage. You have to store it. Thyroid is the only gland which stores thyroid hormone, and thyroglobulin contains about 8 T4 molecules. Then utilization. Thyroglobulin, when there is a need, when the cell is under the influence of TSH, that thyroglobulin is taken up by the acinar colloid into the cell by pinocytosis, and it undergoes hydrolysis, so that T4 is released from the thyroglobulin molecule, and it is, thrown into the, it is secreted into the circulation. So T4 is released in the bloodstream, So during the process, some of the MIT and DIT will remain unutilized and they are dehydrated and recycled because that is a scarce element in those days. That's why the body which evolved for over crores of years of evolution has made a mechanism to preserve the iodine, which was a scarce element in many areas of the world. Then the transport of thyroid hormone, most important part here, clinically relevant because this you must know for interpreting the thyroid reports. So transport of thyroid hormone is occurs by thyroxine binding globulin. 80% of the T4 and 60% of the T3 is bound to TBG, and these are three molecules you must remember. This will be asked what are the carriers of thyroid hormone. That is thyroxine binding globulin, thyroxine binding, this is albumin, and thyroxine binding pre-albumin. You must be able to tell this thing, you must be able to write this thing. This might be asked in the question, as a MCQ question or in your viva. Then thyroid hormone undergoes catabolism in the peripheral tissue. I told you that T3 is formed mainly by the peripheral tissue, not by thyroid. 80% of T3 is produced by peripheral, in the peripheral tissue. And then T3 and T4 are conjugated with glucuronic acid in the liver and excreted through the bile. Finally, it gets excreted through stool, through the intestine. Okay. This is a schematic diagram of thyroid hormone, which you can get it in the book that I told you from this capillary cell. This iodide is taken up. This is trapped in the acidic cell, and it is taken up. MIT DIT is formed, and then T3, T4 is formed here, and finally it is released into the blood to the peripheral for utilization in the peripheral tissue. So this is again summary. You must remember this step, very important step for interpreting the result. See, remember T3, T4 is found in two forms. One is bound form and one is free form. The free hormones is the biologically active hormone. Remember this thing, and that is the prime regulator of TSH secretion and controlling the disease or maintaining normalcy. This you must remember, not the bound fraction. T3 is biologically active and it exerts action and regulates action. Basically, in the periphery, T4 gets converted into T3 and then it exerts action. Basically, the free T3 is the main regulator of its own secretion by the thyroid gland through control mechanism by TSH and TRH. I will tell you later. Now, T4 is formed, as I told you repeatedly, that it is by the thyroid gland, and T3 is formed in the peripheral tissue mainly. So, since T3 is formed from T4, T4 is also called a prohormone. Now, one important part you must remember here, only this point, that the relative metabolic potency. Basically, T3 is more than 3 times active than T4. That you must remember. And that's why nature has evolved a mechanism to convert T4 to T3 so that with each conversion, one iron atom is saved so that it can form one more T4 for further utilization. So, relatively, T3 is metabolically active and T3 level becomes decreased or increased when the disease becomes very advanced. So, this is a prime regulator. This tries to maintain the body somehow to compensate so that the body doesn't suffer from thyroid hormone crisis. The regulation of secretion, I think I know, I'm sure you in the class 12 you have learned this part, that what happens basically, just you remember this thing, that here you look at this thing, that hypothalamus is located in the brain. From here, TRH, that is thyrotropin, that is TSH releasing hormone is secreted, and which stimulates anterior pituitary, and anterior pituitary secretes TSH, and this stimulates thyroid gland to produce thyroid hormone. Now, when the thyroid hormone is produced in excess, obviously that FT3 and FT4 will be also high, and that will exert a negative feedback to the TRH first, to the hypothalamus first, that don't release so much of TRH. So obviously, TSH will not be secreted that much in response to less TRH, and less thyroid T3, T4 hormone will be synthesized. Similarly, if the T3, T4 level becomes less by chance, so that will stimulate TRH to produce more TRH, hypothalamus to produce more TRH, accordingly the pituitary gland to secrete more TSH and to secrete more T3 T4. But the disease process, I mean, once this is the normal mechanism, but that will fail in disease condition. Everything has a capacity, so also thyroid. So there will be a capacity to respond to TSH by the lower limit. Suppose the disease process has occurred and by which the progressively thyroid gland is getting destroyed and T3, T4 is getting progressively decreased. But this, through the negative feedback mechanism, it is stimulating the TRH and finally it is stimulating the TSH. So to release excess amount of TSH so that whatever tissue is available in the thyroid because of the disease process, let it produce adequate amount of T3, T4 to cater to the needs of the body. Okay. Now, so if you find that T3, T4 is less, that means TSH will be high. That means the disease is, I mean there are three stages. See, disease can occur at the thyroid gland. If the disease involves the thyroid gland, that is called primary. If the disease involves pituitary, that is called, in our time these are three terms are used. Now these three terms you will not be able to find in your textbook. For your own information, don't get diverted, otherwise this way you have to interpret. See, sit down, no issue, sit down. See, if the disease involves, I repeat, if the disease involves thyroid gland, it is primary hypothyroidism, primary involvement. That means it is primary hypothyroidism or primary hypothyroidism. If the disease gets involved with the pituitary, that is called secondary. Pituitary can also suffer from have some diseases. Thyroid is intact, and because if the pituitary is involved and there is a tumor in the pituitary, and if it secretes more TSH, obviously more T4 will be secreted, T3, T4, and the control mechanism will be lost because tumor is there. That is not under the control of normal mechanism. Similarly, in the hypothalamus also, there might be a tumor, there may be damage. Suppose a person has sustained a head injury and that hypothalamus is destroyed. Maybe pituitary is also destroyed because of that impact of the injury that you have seen that in the railway accident yesterday. Many people will sustain head injury. They don't know what is the degree of damage. In the degree of damage, either for the accident or because of the damage, these glands can be involved, or because of the disease process that can have a tumor and that can secrete excess amount of hormone. So that is, so try to remember if the disease involves here, that is called secondary, and that it is involved this hypothalamus, that is called tertiary. Now again it was during in last another 10 years there are two terminologies which are used. One is primary and one was central, because these two were coupled together. Secondary and tertiary used to be coupled together to central. Now again the nomenclature has changed. Now the tertiary and tertiary part is withdrawn. The central part is also withdrawn. Now in the latest book that part I am telling you, the latest classification which you will be reading in your 4th year class. Latest Harrison book is available from where I have copied it. I will show you that central part is not there, only primary and secondary hypothyroidism is there. So some clinically relevant information here you must see. When you find a normal thyroid report, please try to say euthyroid status. The report is euthyroid and not normal. We are very much habituated to say normal. If you say euthyroid, it has a meaning that you know the real terminology. Now, if it less functioning, that then we say it is hypothyroidism. And this hypothyroidism is the most common disease. Whatever be your specialty, every day in every clinic, at least then if you get 100 patients, 5 patients will be diabetic and 1 patient will be thyroid. And usually these are hypothyroid patients. So that is also the bread and butter of all the endocrinologists. So you must know the hypothyroidism in detail. Hyperthyroidism means more functioning thyroid gland, more thyroid hormone levels. Basically, we know the status of thyroid functioning by drawing sample from the blood at a given point of time. Thyroid, this hypothyroidism and hyperthyroidism are detected in the patient, but relatively in a very advanced stage. The symptoms doesn't occur, patient doesn't come. That's the problem. And whatever be the clinical diagnosis, be it hypothyroid, be it hyperthyroid, I mean functional status, I repeat, it is not tumor, it is not cancer, it is not otherwise cyst, otherwise. Only thing is that you know to know whether the thyroid is functioning normally. I repeat, not structural changes. Whether it is normal functional changes. No, I am repeating repeatedly, not structural changes. Forget about the structure. That is for that another investigation is required. Whether thyroid is functioning normally or less or high, that is basically thyroid function test biochemically. So once the disease is diagnosed, even clinically, you require a biochemical confirmation. And biochemical confirmation is required because if you start the treatment, you have to monitor it. And biochemical report is, almost mandatory. Without biochemical confirmation, no clinician is going to start the treatment. So detected biochemically in the very early stage before in the symptom and sign. Suppose if I want to make a T3, T4, TSH analysis or FT3, F4 analysis in this entire population, maybe about 300 patients, 300 students, first year, second year, maybe 1 or 2 might be detected to be suffering from subclinical hypothyroidism. That is subclinical, means detected biochemically, just like blood sugar. At least 1 or 2% might be present to be suffering from diabetes mellitus or maybe impairment. Without that thing, you cannot do this thing. So that is the purpose of, that is the uniqueness of thyroid hormone, biochemistry, and by measurement of thyroid hormone in the blood, you can detect in a very early stage before the disease becomes evident. So this is a stages of disease. This is not written anywhere in the book. This I have realized over last 30 years of time, that what is the basis of and what are the stages of diseases and why biochemistry or biochemical tests are unique. See, gene-environment interaction is required. That is the basis of any disease. The first is genetic defect. Genetic defect might occur at birth or it might be acquired later after birth. Means there might be some chemical exposure, there may be radiation exposure. If somebody gets exposed continuously to radiation, obviously DNA damage will occur. And that person is going to suffer from some kind of genetic disorder, genetic problem, and most commonly cancer occurs due to exposure. Similarly, thyroid also can be involved also, or any other diseases. So genetic expression, because the genetic defect is present at birth, but it gets expressed at different time interval periods of life. Some diseases get at birth, something at early infancy, childhood, then adolescent period, then adult, maybe in the late age. And then biochemical changes usually occur at the molecular level and within the cell, and that can be detected by biochemical testing only. There is no other means. If that stage is allowed further, then obviously the cellular metabolism will be altered and cell will undergo changes, and then you will find the tissue changes. At this stage also, this disease cannot, there will be no sign and symptom. But when the patient presents with the features, that is symptom and sign, that is too late, and usually too much damage has occurred and the disease is a little bit advanced. The more you find symptoms, the more the advanced the disease is. So this you just remember, I thought that I must share this information. You just remember these 4 steps. Without these 4 steps, no diseases occur. At this stage also, after these pathological changes, radiological changes occur. If you, because the disease is there, but it can be detected only it attains some amount of size. Even by MRI, it requires some amount of size, small, very small size, which is detected pathologically by the microscope, cannot be detected radiologically. And there will be a mismatch between the pathological finding and biochemical finding. Why there is a mismatch? Can you now realize? The clinical is little bit advanced before the radiological, and then pathological, and then biochemical. So this, there will be really not matching what doctors expect. There will not mismatch because of this thing. The size is not matching, symptom is not matching, pathological report is not matching, the report is not matching because of this thing. Somebody might be having little symptom, but on pathological biopsy, there will be grade 4 cancer. So that you have to accept it, whatever be the symptom. So goiter, basically any enlargement of the thyroid gland is called goiter. I am telling you a little bit which is not relevant right now at this stage, but maybe it will be very much useful in your clinical practice, because after all you will be doctors. So from the first year itself, I am trying to tune itself so that you can interpret the report and get clinically oriented, not pure biochemistry, Because biochemistry, I know very well that biochemistry, physiology, anatomy, all these things are boring subject to read. But to make you a little bit interesting and clinically oriented, I have incorporated deliberately some of the topics which might make you interesting. One is goiter, because this you will see in the clinical practice. Any enlargement of the thyroid is called goiter. Where there is iodine deficiency, I mean not iodized salt is not available, geochemically that area is iodine deficient, so that is called iodine deficient goiter, and that is called an endemic goiter. There are still several areas where endemic goiter is still prevalent, and these are called goitrous belt. All areas are not developed like this. Don't try to think that Bolpur and West Bengal or other places are everywhere is situation is like this. And where iodine is sufficient, here also goiter occurs, and this goiter which you will see, and that is autoimmune, because of the autoimmune diseases. And the goiter which you will see will virtually belong to autoimmune variety, not a deficient variety. Okay, that you must remember. So because of the widespread concept that despite taking iodine, people used to suffer from goiter, and believe me, in the 90s, doctors were also under the impression that if the patient presents with goiter, that it is due to iodine deficiency. So what they used to prescribe, that is Lugol's iodine, that contains a very high amount of iodine. And patients are also, and the media also, used to spread the message that iodine deficiency causes goiter. That forced them, see, this is the actual goiter which you will be seeing. I have seen during my MBBS class this type of goiter. And you might see here also, because I'll show you a video and that will be clear. See, you might see these are all advanced cases. Normally, nowadays, you will see goiter of this stage, because everybody now looks at the mirror almost daily. So if there is any enlargement is found, so you will detect the disease at a very early stage. So your eyes also must be tuned that when the patient comes to you and look at you, this part is open, be it male, be it female, you will have a look, you will be able to diagnose that whether thyroid gland is enlarged or not. So because of the widespread belief that iodine causes, second slide I am telling you. See, goitrogens are, that is basically substances that prevent utilization of the iodine. We must know that we are taking iodine in the diet, but that iodine is not getting absorbed properly by the intestine, or maybe by the thyroid tissue. The reasons are many. There are many goitrogens which prevent utilization of the iodine. It is present in the water, it is present in the food. Because if you look at the Damodar River, it is passing through a coal-rich belt. So that Damodar water was contaminated with coal-derived pollutants, and a number of industries are located across the river Damodar. So is also Ganges. So these are the phenolic compounds. You can see coal-derived organic pollutants that usually contaminate. I'll give you this book. You will read. This you will read at the clinical stage. This is too difficult topic. I'll share this thing at an appropriate level if you want to show, sir. Just to tell you that you are taking iodine, adequate amount of iodine, but because if the water in that particular area has a high amount of these compounds, that iodine will not be utilized. Obviously, artificial deficiency of iodine. What I want to say. Now, many areas, many people are taking well water. So if that got contaminated with bacteria also, so that will also produce some amount of goitrogenic substances. Now, normally in the food also, which are commonly take, that also cause goitrogens, like cabbage, maize, millet, sweet potato, beans, and mustard seeds. Common Bengali foods or other Indian foods, this include. But see, remember, the amount of cabbage which we take is not that much. Just we don't take cabbage. We take cabbage only hardly during winter season, maybe once or twice or thrice in a week in one vegetable. And that is a small amount. Fortunately, now cabbage is available throughout the year, or everything is available throughout the year. But we don't take that much amount of cabbage, which will cause goiter. Okay, there is a balance. Everything is present in that thing. So if you can say we are taking cabbage, that can cause goiter, no. Answer is no. One must be food fetish. If we take that cabbage morning, evening with every food, then you are taking excess amount of cabbage. That can cause. Okay. So because of that belief also, that iodine content deficiency causes this thing, goiter. So doctors used to say prescribe also iodine, and patients used to take also seafood to get more iodine, and they used to present with more goiter, and there is a considerable abnormality in the biochemical findings. That's why addressed to the Indian Medical Association gathering in its annual function in 1998-99, to a number of doctors that don't give iodine, these patients require thyroxine, and I explained why the iodine is not being utilized properly, and this patient iodine will not be of any help. So goiter, I told you that it is long known with primary hypothyroidism, that is iodine deficiency, and iodine, that is endemic goiter. This is not an endemic goiter which we see today. This is autoimmune. I repeat, this is autoimmune, not endemic. So not found nowadays because dietary supplementation of iodine, iodine salt, maybe with other foods. Occurs in hyperthyroidism also, occurs in euthyroidism, compensated state. Compensated means suppose somebody is having iodine deficiency temporary, or there is a temporary deficient utilization. Just like you have gone to somewhere else, you are spending some days where that water is contaminated with that goitrogens. This is preventing some iodine utilization, and for the time being, you have produced some less amount of hormone, and because of that, TSH was stimulated, and that TSH, stimulated thyroid gland to increase. So that caused some amount of goiter which will might regress. So in euthyroid thyroidism, also you will get, goiter. But in that case, if you measure the T3, T4, T6 level, it will be normal. So goiter might be associated with euthyroid, primary hypothyroidism, not central. Remember, if there is any involvement of the pituitary gland or thyroid gland, the gland is not going to enlarge, because problem is at the pituitary level or hypothalamus level. And hyperthyroidism, thyroid tumor also causes thyroid cancer. These are not our topic. Again, I am telling you, repeatedly in this class, we are trying to know the functional status of the thyroid, not structural changes. The diseases of thyroid, this is the classification which you will read in your clinical years, in maybe in next year or later. This is the classification. You can see that tertiary term is not here used, only primary and secondary. So that tertiary page I told because we were taught, and that is easy to remember to indicate hypothalamus. So you will read all these things. I am not going to tell you in details, but this is the main classification which you have to write in your clinical class. There is no excuse, and this will cover the entire things in your MBBS curriculum. So one most important thing I must say, because congenital hypothyroidism. You see, if the babies are born with an iron, defective iron, T3, T4 synthesis, the baby requires immediate urgent screening, because if you allow these babies to grow a little bit, they will have a permanent mental retardation. And anything, no family wants, no mother wants a permanently mentally retarded child. That's why nowadays TSH test has become mandatory soon after birth, almost, to detect this thing, that if the baby suffers from hypothyroidism, thyroxine hormone has to be given to prevent brain development properly and growth development also properly. If brain development doesn't take place properly, and if the child is detected a little later, he is going to have a permanently mentally retarded child, which is really a botheration. So please make sure that if you are a pediatrician, if you get involved in delivery, get the sample drawn and get it tested also. So permanently retarded child can be prevented by a simple blood test. So coming back to thyroid function test. So in reality, biochemical thyroid function test is not actual real-time test. Like in the glucose tolerance test, what we have given a glucose amount, and then at half an hour interval or at 2 hour interval, normally we collected a sample. What is real time? Suppose today we have the person has taken glucose at 9:15 and we have collected a sample at 10:15. That is a real time. And if you want to make a GTT, then a real time 9:14, 9:45, 10:15, 10:45, 11:55, 15. Okay, that's a real time, how the body is handling. Here we are not really assessing that functional status. So we are drawing a sample at a given point of time. Today at 9:15 the sample was taken, and what was the level of thyroid, that is T3, T4 and TSH or FT3 and FT4, that much we are knowing. So that indicates an indirect assessment of how the thyroid function is today, and definitely it is reflecting some activities. Actually, I repeatedly tell you, we are assessing the thyroid function state, not the structural changes. These real-time function tests are done by the nuclear medicine department. As I told you that if we take a dietary iodine that is taken up by the intestine and it is taken up by the thyroid gland. Now here what is done, a radioactive capsule containing iodine is the patient is asked to swallow. The patient takes that radioactive capsule. After 6 hours usually a radioactive scanner is placed on the thyroid gland and it is detected whether the thyroid gland has taken up radioactive iodine or not. If the gland has not taken up, then that means thyroid is not functioning, it is not taking up. That study has to be done, and that is done by the nuclear medicine department. And you can see this is called a thyroid scanning. Now you can see, you just look at the thing, this is a normal, and if there is a mode of uptake, you will get a picture like this. Okay, so if there is a toxic mass, there is a tumor, you will get like this. So these structural changes are not part of today's discussion. Again, how much radioactive iodine, whatever, suppose it is 100%, how much is taken up, you can see this is 2%, this is 50%, this is 45%, this is 70%. That is also calculated. I mean, one must be able to know this, interpret this thing, what is normal, what is hyper, and what is hypo. That's a real-time testing, real-time functional status testing. Ours is also some kind of real-time also, but it is not of that magnitude. Now, biochemically, as I told you, these only 3 parameters are done, T3, T4, TSH, and fortunately or unfortunately, only these 3 parameters is done also in Santiago Medical College today. Even today, FT34 and FT4 is not done. But if you are allowed, if you are given a choice, forget about total T3, T4, concentrate on FT3, FT4. Okay? Why this is important? Why total 3 T3, T4? Because I told you that FT3 and FT4 is the biologically active hormone. So we have to see the status of the biologically active hormone. What is the concentration? See, if you measure total TBG, what is the, see, another most important thing, you will be seeing all the biochemical parameters. See, be it cholesterol, be it glucose, be it urea. And if you look at the lower limit and the upper limit of any individual parameter, the difference will not be more than 3 times, 2 to 3 times. You can look at this thing, T3 level, it is 2.5 times, 0.8 to 2, it is 5.1 to 14.1. In contrast, this TSH is the only hormone which is the minimum range difference between the two is 15.5 times. Even there are many laboratories which report that normal interval, which is about 20 times. And no other parameter, biochemical parameter, is having such a wide range that means 2.7 to 4.2 means if 0.3 is also normal, 4.1 is also normal. Can you imagine the degree of difference? So that means it's a wide, I mean, there is a very wide range. The main reason is that the relationship between the FT34 and FT3SH is inverse log linear. That is the main reason. If you want, I can clarify this thing in the later stages, but not at this stage you require. Only thing is that I wanted to highlight here that TSH is the only parameter where the difference between the two, lower limit and the upper limit, is highest. So these tests are done in serum. Never ask the laboratory to do the test from the blood. So serum T3, T4 and TSH, it is assessed the thyroid function. I have been repeatedly telling. You want to go home, you can sign with Regina and then you can leave. You will be missing basically an important part at the end. Anyway, you just move ahead. To assess the functional status, whether normal, that is euthyroidism, hypothyroidism or hyperthyroidism. Now, you will not get all the facilities everywhere, and all the people will not be that much economically supportive. Suppose you might be having a choice, you have to do a single test. A single test, if you want to do, go for TSH, simple TSH estimation to detect euthyroidism, normal or hypothyroidism. That you must remember. Everybody cannot afford money. So if the financial condition is, I mean, very bad, please go only single test, that is TSH. but a normal level excludes the primary involvement, which is the major involvement, that is exclude function. Limitation of PSH estimation, that is very, very rare, and since this is used in newborn screening, only thing is that if the newborn suffers from severe non-thyroid illness, that can cause alteration and you can make a false diagnosis also. And if there is taken a secondary involvement of the pituitary hypothalamus, you will not be able to diagnose this thing. Free T3-4 and T4 corresponds to the biologically active pool, as I have been repeatedly telling you. And if you are given a double test for confirmed hypothyroidism, go for TSH and FT4. And if you want to detect triple test, if you are given a choice, then okay, no problem, you go ahead. Then you choose for TSH, FT4 and FT. This is a practical problem I am telling you. This is you have to face this thing. Because there is an entity where FT3 and TSH and FT4 might be normal also. But if you don't do FT3, you will not be knowing whether this is an FT3 toxicosis or not. There is an entity. That's why it is better if you think that a patient is suffering from hyperthyroidism, which is relatively uncommon, go for TSH, FT3 and FT4. And if you want to know the complete status of thyroid function, get all the 5 tests done, which I normally do. And if you explain the patient properly that I want to know, at least before the start of the treatment, I would recommend that you somehow convince the patient to undergo these tests, and usually patients comply despite his clinical financial condition. At least this test you've done, next time I will not allow you to do this. So that as a clinician, you know the baseline picture before starting of the treatment. That point will be lost. Never in a one time that point will not come back. So whatever investigation you want to do on any patient, before starting any treatment, do all the tests whatever possible. Because that stage is will be missed throughout your life, and you will not be able to know what was the status at that point of time. And that will give a very good prognostic figure. But you can see also the future of that patient, that what is going to do, you are going to suffer from. The laboratory test, total T3, T4, if you do, what is the limitation, you will try to understand. It is mostly protein bound. As I told you, 99.97%, 99.0. You have seen that free fraction is only 0.3 or 0.03%. It is mostly protein bound. So any factor which increases TBG level will increase total T4, not the free T4. So common condition is pregnancy, estrogen. Most of the women during the fertile period take oral contraceptive. These are the 2 conditions you must remember and inquire when you get an isolated T4, T3, T4, normal, T3 and TSH is normal, but isolated T4 is elevated. You think that whether the patient is pregnant or whether his patient is taking oral contraceptive. If the patient pregnancy is advanced, you can definitely see by seeing the patient. But if it is an early phase, you will just inquire. And patient usually gives a reply in affirmation that yes, I am taking oral contraceptive. And similarly, if the total T3, T4 is low, but TSH is normal. So that means that is due to decrease in the TBG binding, which can be due to androgen nephrotic syndrome. Nephrotic syndrome, you have seen that heavy proteinuria takes place. So along with that albumin, along with the TBG, everything is getting lost. So because of based on that thing, that TBG level, that T3, T4 will be less also. So TBG level measurement is another factor. Then you have to measure TBG level. So it is not done. So we have to assess by taking the history. So this is a peculiar situation. If FT3, FT4 and TSH is normal, total T3 and T4 is high or low. That's what I exactly told. That you take the history, whether the patient is having pregnancy, whether the patient is taking oral contraceptive or other drugs disease. Main cause of high T4 and T3 is increased TBG due to any cause. I repeatedly told you must not forget this thing. Similarly, if the total T4, T3 is low, there is decreased TBG level due to any cause, albumin level or drugs displacing T4 and T3. Now, since you will be handling autoimmune thyroiditis, autoimmune goiter, if you diagnose a case of hypothyroidism, don't forget to measure anti-TP antibodies. That is an essential part of the investigation that you must do it. Once the patient is diagnosed, you go for anti-TP antibody, because the importance is that almost all patients with hypothyroidism, up to 80% of patients with Graves' disease have TP antibodies at high levels. But if you see the normal individuals, see 5 to 15% of the euthyroid women suffer from thyroid disease much more than men, and up to 2% of the euthyroid men have thyroid antibodies. Now, thyroid follicle contains globulin, thyroglobulin. If the patient suffers from thyroid cancer, obviously that follicular structure is destroyed and thyroglobulin is released into the system. Now, after the cancer is treated, be it surgery, be it radiotherapy, be it chemotherapy, the patient needs follow-up. And during follow-up, it is important to see whether the cancer has recurred or not. Just to see the recurrence, the earliest parameter. It cannot be detected even by, I'm telling you, even PET CT or MRI, anything else, whatever you do. The earliest sign of recurrence of cancer will be an increase of the thyroglobulin level in the blood. So all patients coming with a cancer, thyroid cancer follow-up, do in every visit they require thyroglobulin estimation. That is the importance of thyroglobulin, serum thyroglobulin. You must not forget this thing. Thyroid cancer you will see less, but those patients, rare patients you will get, you must forget, don't forget that the earliest sign, I would say, is increase in TBG level. Means cancer has recurred. You have to take adequate steps. So another hormone, another test is there, that is thyroid stimulating antibody. This is receptor antibody. It is not done basically, it is done in the research center. So importance is this test, as I told you, it's either to confirm and exclude if your clinical diagnosis is hypothyroidism. Now after you have started the treatment, you require monitoring. Whether the patient, the disease is under control or not, whether the patient is taking the dose also or not. That is also your headache. The patient will usually tell a lie. That is another factor. And a disease modification. I am telling you, I have seen a case, I have been the one patient I have come across, and he is under my treatment, she is under my treatment for last 6 years, I could not die. I mean, the disease is fluctuating like anything. That is a hyperthyroid case. I will share you the video. The methods of estimation, only you know the terminology only. There are 3 methods which are done for measuring thyroid hormone. One is radioimmunoassay, that is the gold standard even. Now, enzyme linked immunoassay, that is done something other medical college nowadays. So they during your internship, the report we will get will belong to this test. It's enzyme linked ELISA and not CLIA. CLIA is done by the chemical immunoassay is done by usually by the big laboratories. Maybe during your internship or housekeeping period, you might get CLIA also in this hospital. But CLIA has come in India from 1998. So if you want to know RIA or ELISA, you have to refer principles and then read the literature. Now, while interpreting, this is now you just come to the main point. You have to do this exercise. While interpreting, first look at the TSH level. The moment the patient presents with the report, look at the TSH level first, whether it is normal or high or low. Then FT4 if it is done, else T4, whether it is normal, low or high, then FT3. Because this is, I mean, affected at the end. So FT3 will be lower only in advanced cases of hypothyroidism. Similarly, FT3 will be high in case of advanced hyperthyroidism. So this you must remember. Now, interpretation will be that in euthyroidism, all will be normal. In primary hypothyroidism, what happens? That T3, T4, FT3, will be normal and TSH will be only high. As the disease progresses, later stages, first FT3 and FT4 will be low. And then total T3, T4 will be low and TSH will be very high. Hyperthyroidism in the early stage, FT3, 4 will be normal, TSH will be low. And later stages, again FT3, 4 will be high and then T3 and T4 and TSH will be very low, non-detectable. So this is a child which was born from, I have been following up from 5 years of age. You can see here, this is a real time. I got this video 3 days back from his father. I mean, I told him to get a photography. You see, this is a child when he was, I mean, almost 5 years of age. There is no increase in the gland. Now you see there is an increase in the gland. Can you see? Can you see the gland has increased? Because her mother was suffering from a goiter, and I delivered a lecture way back in 2001 at Sanjay Gandhi Postgraduate Institute of Medical Sciences to DM Medical Genetics students. where I told the genetic, gave a lecture for 1 hour on genetic factors of thyroid diseases. And because of the knowledge, I thought that since the mother was suffering from hyperthyroidism, mostly the child is like, there is a high chance that child can suffer from hyperthyroidism also. So I followed it up, and this patient was detected at an early age. This patient is under my treatment. The disease, this still, sometimes it is increasing, sometimes it is decreasing. I'll show you the video of this thing. Formally it is coming to an end. Just this same child. If you want to see thyroid, ask, give him a little bit of water, ask to deglute it. Can you see? Can you see? This thyroid, small thyroid, this is normal almost. This is not enlarged. Always you give a little bit of water so that she will be able to deglute it properly. Because I told casually that you just deglute it in front of the. Now you see properly, look at the very closely. Can you see the increase? Can you see the gland? Thyroid gland is moving. Can you see? Just look at this thing. Because periodically you need to follow it up, and I would say because of the very easy availability of mobile phone, to tell the patient to record, take a still photograph and a video also, keep a date, so that during follow-up you will be able to know what are the things. If the patient doesn't have that facility, at least spend some time to, some money, I want 10 rupees money to give a DVD also. Don't matter, it will not cause any harm to you, but it will be of real benefit to that fellow, to that patient. Every time you ask them to bring that CD and you get it copied, nothing to help a poor child who doesn't, poor parents who suffering from a disease. Usually I make always a recording, okay? Now coming to the last part, that is you have to now interpret, okay? Now you will not be, you have seen, you have learned this thing, the patient has come, and now you have to interpret, okay? This actually I have taken a very clear cut, very advanced cases of, from your laboratory, old laboratory. This is the actual report. I have deliberately blocked the name of the patient to block the identification. So what is this case? Because the patient will ultimately come with the report and you have to interpret. And all of you have to tell before you are allowed to leave this class today. Now what is this case? Tell me. You can tell something wrong, you need, I want to rectify. You just tell, yes, you are trying to, last bench, you just tell something, no issue, nothing, please stand up, at least I have seen you, you are trying, just you will be able to do this thing. Huh? What is the diagnosis? What is your diagnosis? I have been telling you. This I am not asking you to interpret. Now you have to tell a diagnosis. I have been telling all these things. If you know the regulation, if you know the understanding, you will be able to tell. It is a very overt case, very advanced case, I repeat. Why are you fumbling? Is it why this is it is coming? Yes, it is hypothyroidism emphasized. Is it not? No, that is not is it. She attempted, you must congratulate that she answered also very correctly. Now you explain. You explain. Okay. No, don't see, everybody will be given a chance. At the end of my lecture, always be try to attend it, that you will be given a practical problem and which will be relatively overt. At first year level, I don't expect that you will be able to, diagnose a complicated case. That's why deliberately I have chosen, and this is an overt case of hyperthyroidism. You explain to your batchmates, why did you diagnose it hyperthyroidism? First, what I told you, first you look at the level of TSH. What is the level of TSH here? Is it low, normal or high? Normal. More than normal. Is that finding? And peculiar, now you have told it, I mean, by guessing, now I understand. What is the normal range? If you don't understand, you can tell me what is the normal range? And what is the level the patient is having? This is the patient level. And this is the normal level. It is low. TSH becomes low in which condition? Hyper normal. You just tell. That part you must be clear. Hyper. Hypo, it will be high because I told you if there is a decreased synthesis of thyroid hormone, that will give a feedback inhibition, feedback stimulation to the TSH, pituitary gland to secrete more TSH. So TSH will be more. But if the gland produces excess amount of thyroid hormone, that will inhibit TSH, thyroid gland, not to release any hormone. And if the disease is severe, a little bit allowed to be aggressive, this figure will go to suppose 16.14, and this will come down to 0.01 to 0.015, and then at one point of it cannot be detected within by the laboratory. I have seen more than 20 also in my life, and their TSH were undetectable. So you guessed something, but you could not answer properly. Okay. So anyway, you have attempted. I must congratulate. And there T3 and T4, if you look at the value, it is high. Because it is high, it has suppressed thyroid gland not to produce any TSH. And the disease is relatively advanced. Little bit, not more too advanced. If too advanced, this might be 20, this might be 24, we don't know the figure. And this will be, in that case, it will be non-detectable. If that thyroid pituitary hypothalamic axis is normal, remember, that means there is no disease in that organ. Okay? Second case. What is this case? Sit down. You just tell from one side here, just attempt. You just attempt. Stand up and try to tell. You try, commit mistake, I don't mind. I don't mind to try. I have told you and you must be able to interpret. What is the TSH level here? There is no TSH, that means there is no hypo, not hyper. This much you must be clear. What you are getting here is an isolated elevation of T4. This is only T4, total T4 is elevated. I told you in which condition total T4 gets elevated. T4 or T3? You tell me in which condition total T4 and T3 is increased. Hyperthyroidism is another cause, but here it is not supportive. The camouflaging type of report. When you get a normal TSH level but increased T4 and if you also increase T3 also, you think, just look at the gender of the patient. It is a female patient. The probability is that the patient might be having pregnancy. If she is not pregnant, she might be taking oral contraception because she is in the fertile period. So these two will simply inquiring these two questions will clarify the doubt and the patient will be satisfied. If you don't clarify this properly, then there will be thought inside the throat. The patient will continue to say, my T4 is high, my T4 is high, my T4 is high. And doctor will erroneously might give also, if he does not properly, anti-thyroid drug, which is not desired. If you are not sure about diagnosis of anything, don't try to prescribe drug. This is a clear-cut example. Deliberately they have taken this from this female, that is 26 years female, not from other age group. So whenever I repeat you, whenever you are seeing a normal T4 level, TSH level, but T3, T4 level is high only, you just look at the age and the gender of that particular patient. If the patient takes, thyroid oral contraceptive for a prolonged period, or if the pregnancy is suppose in the third trimester, almost the patient is going to turn, is going to deliver a baby within another 1 or 2 month period, then T4 will also be high, T3 will also be high. but at that time high TBG level will be there. This is an early phase, you can say, earlier phase, maybe in the first trimester of pregnancy, or maybe she has been taking oral contraceptive for maybe 3-4 months or 6 months. That's why only T4 has been affected. Okay, that is the reason why a small amount of TBG has been elevated. Okay, what is this case? Some of you just try to stand up. You try to understand, explain this thing. No, you try. You just try. See, you have to interpret ultimately, finally. That is what is my purpose. I could have stopped the class by discussing this thing, but this is a practical problem we have to face. Huh? বলো জোরে বলো, ফোকাস করো দিকে, ফোকাস করো। Hypothyroidism, কি জন্য hypothyroidism বললো বলো? Yes, you have correctly said. আমি বারবার বলছি কি first look at the TSH level. I have been telling you repeatedly that look at the TSH level first. What is the TSH level? It's abnormally high. It's very high. That itself diagnosis, even if you don't require any T3 T4 test in this case. T3 T4 is required. Why? Because I am telling you why it is. If there is a pituitary tumor, that can also cause TSH elevation. In that case, T4 and T3 will also be high. If the pituitary has a tumor and that is secreting excess amount of TSH, of course this TSH will be high. But in that case, because of the hyperstimulation by the thyroid gland, this T3 and T4 will be also high. That will cause secondary hyperthyroidism, not hypo. This is primary hypothyroidism. I repeat, it is a primary hypothyroidism because the hypothalamic pituitary axis is normal, and because of that thing, I mean, due to remarkably lower limit, you see the lower limit is only 4.5 and the level is 0.83. The level is too low and pituitary has been stimulated almost maximally, and it can go further down, even 0.3, then TSH level might be again 500, 600. And I have seen one child presenting to me, I mean, with the TSH level of 1700, 1700. So the more the degree of, I mean, level of lowering of the total T4, if you see the FT3 in this case, FT3, FT4, it will be remarkably low because total T3 has gone down. You understand why I am telling? It is relatively advanced case. বলো যে বুঝতে পারেনি বলো, whosoever has not understood I will explain. That is if see, don't get confused. I do not want to confuse you. Suppose if we don't do any T3 T4 here, we have blocked it. Okay, I told you if you have given one test that you do TSH. Now you are doing a TSH. Now if a TSH is there, that means it is a primary hypothyroidism. Okay. Now there is also a possibility that pituitary gland also can increase. I mean, because of a tumor, that can also increase high amount of TSH, and that will be unregulated. I told you this is normal, not normal. So if TSH is secreted in unregulated amounts, then that will stimulate the thyroid gland to produce more T3 and T4. With this TSH level, T3 level will be almost 12.5, it might be 32, 42, we don't know the figure. But what the maximal capacity of the gland, that the gland will go on secreting. Similarly, T3 level is 1.8, it will be 18, 20, 30, we don't know what is the figure. So in that case, that is a secondary hyperthyroidism then, not hypo. If you know the regulation very properly, as I told you, whether the problem is at the thyroid gland or whether the problem is at the pituitary or hypothalamus, that is secondary. Pituitary and hypothalamus is secondary. if the gland disease involves the thyroid gland, which is most common, think the common things first. Don't think the uncommon things. Then you will miss also the diagnosis rarely. I mean, you will commit the mistake. Pituitary tumor, hypothyroid tumor is not that common. You will find other features also in that case. Okay. Is this point clear? Because of the remarkably low T3, T4, thyroid gland, this pituitary gland has been stimulated very much, very high, and that it responded by producing excess amount of TSH. Okay, that means excess amount of TRH was also there. Hypothalamus also was stimulated because that released. The TRH test is not being done. If you do the TSH, there is no need for TRH also. In this case, if you want to do the TRH will also be high. But without hypothalamic axis, this will not function. Thyroid gland is under the influence, this pituitary gland is under the instruction of hypothalamus. The hypothalamus is diseased, obviously, even if the level is low, you will not get high TSH level. That you must remember. Is this part clear? You just look at this level. What is this report? You tell, every one of you have to tell. Now from this side you tell. What is this report? You tell. A was normal. He is telling normal. Do you agree? You are telling also normal. What is your opinion? Normal. What is your opinion? You who are in favor of normal, hands up please. Hands up please. Who is normal, in favor of normal? Who is in favor of normal? Okay. Only few, some half. Please hand up fully, those who are agreeing. Yes, you did not, those who have not raised, what are the issue? What is the issue? Those who have not raised, what is the doubt, confusion in this case? Those who have raised, they have correctly said it is normal. Absolutely normal report. So what is the doubt in you? Just let me know. I'll try to explain. Those who are not yet clear, just let me know what is the problem. Huh? Tell me what is? What range? Which range? TSH. TSH is 0.25 to 5. The TSH level is 2.19. You see the level here. It is exactly in the middle. Huh? Male, so what is the problem? The level is normal. Why are you concerned about this thing? Level is, when it is normal, you don't require to do that exercise. When the level is T4, T3 is high, then only I told you. Is it clear? Is it clear? This TSH level is normal. TSH level is normal. That means you need not worry now. We have to look at the T3, T4 only, whether it is normal or not. Is it not normal? Is it not in the normal length? I mean, what is the doubt? I am not clear. Why hello, you tell me. What is the problem? Why you could not understand that this is a normal report? You tell me very clearly. I don't mind. Age borrow reference is not 4.5 to 12. This is normal level. This is how you will get the report. This is the actual report, laboratory report. Huh? Biological reference it only is normal range basically. কত আছে এখানে 4.5 থেকে 12 ফিমেল রে বাবা লেখা আছে ফিমেল এগুলো প্রেগন্যান্সিতে লেখা আছে ফিমেল লেখা আছে অ্যাডাল্ট ফিমেল ও এখানে ওদের বলতে হবে দাঁড়া বলতে ওদের বলে দিলাম লাউডে থেকে আমি অতটা লক্ষ্য করিনি আমি অ্যাডাল্ট দেখেছি ছেড়ে দিয়েছি ওই ফিমেল সে নরমালি বেসিক্যালি ফিমেল অ্যাডাল্ট মেল ওদের লেখা দরকার ছিল ফিমেল অ্যাডাল্ট মেল আর ফিমেলটা লেখা দরকার ছিল ঠিক আছে I'll tell the laboratory to make changes. I'll tell. See, what I'm telling you the reason might be, as I know from a laboratory person, the reason might be since the adult is having the same level almost, that's why they have not mentioned. But specifically, women suffer, have pregnancy, that's why they have indicated favorable. Male should have written, I must agree with you. Male oblique female, then you touch the male clear. I'll tell, no issue. হ্যাঁ মেয়ের থেকে আরে বিভিন্ন ল্যাবরেটরি রিপোর্ট বিভিন্ন আমি বলছি শোনো না দি মেন প্রবলেম ইন বায়োলজিক্যাল রেফারেন্স ইন্টারভ্যাল ইজ বেসিক্যালি কুক ডাটা if you find 5 laboratories you will find biological reference value why in problem is that this reference value has to be found out in a geographical region that is not done regions are coming in the region literature that value is there and they are putting this value simply ওই রকম ওই জিনিস ওই একই রকম লেভেল থাকে টিএসএ সেইটা এরকম থাকে এই একই থাকে এই লেভেলটা যেটা দিস রেঞ্জ হুইচ ইউ আর সিইং if you look at other laboratory I will show you immediately one lal pathology laboratory report that range is also not matching there will be some differences because they have suppose this region they are purchasing from which is made in India they are purchasing regions from Germany somebody is purchasing regions from USA somebody is purchasing regions from Japan whatever value is coming in the literature they are simply putting that value in that test that is the main problem unfortunately we tried to have an Indian data we don't have it is not possible also 1-2 more case and the case will end so this is another case you have to tell what is this case what is this case see other batches I have seen they have very categorically this is early hypo and the other cases they have said that is advanced hypo there is a difference between your batch and the other batch the moment I showed somebody told that is an advanced case and in this case the moment I said that this is an early case of hypothyroidism really an early case of hypothyroidism because T3 and T4 is normal you see again in this report the same report I'll tell them that you incorporate male behavior. Now this is another report FT3 and FT4 this is from Lal Path lab the patient is under my treatment now you can see